Alrighty, welcome guys to the DM module. I do apologize for not having a review session earlier. The T-Force did have an exam the week that you guys finished up week one. And I know week one is a bit traumatizing for a lot of us who went through the DM module. It was a lot of biochemistry that they just threw at you, expecting that you guys would know it and be very proficient in it in a matter of seconds in medical school terms. But trust me, the material is very much, pardon my pun, digestible and understandable, and you will be able to apply it with multiple revisions. And I'll point out the pathways that are more high yield and the things that you guys need to know front and back versus the ones you just need to know very specific enzymes or factors to keep in mind when going through one, your practice questions, two, when you guys are prepping for your exam. Okay, so today we're going to cover DM week one and week two. I know it's a lot, it's close to 300 slides, so pray for me and your guys' soul, and let's do this. Okay, before we kind of get started, can we take a moment to appreciate its pathways, guts, and glory for DM? Okay, sorry, I had to create some corny pun to start off the entire slides. Okay, as always, here's a TPLG with a group of folks, and we are going through a rebranding phase. So if you guys see some updates in the future, you will know. To join the Facebook group, I suggest everyone does get Facebook because a lot of good resources and DES drives are out there. They're fantastic and they help you guys get good student peer sourced material, and it's fantastic. We use them all the time. And the link to our page is you can scan the code and it'll take you right there to Facebook. Okay. All righty. So let's get started. Biochemistry. Okay. So I'm going to cover glycolysis, TCA, ETC, and gluconeogenesis. It's a lot, but I'm going to give you guys the high yield focus so that you can go in and do the reviews that you guys need to make sure that you have this down solid before you show up to the exam day. Okay. Alrighty. So a little bit of first aid to kind of get the ball rolling. These terminologies, if you have a good understanding of them, such as what you need to know for your kinases, right? Your kinases, they help catalyze and transfer phosphate groups, right? If you know that, especially, components such as ATP, it's going to be really important. Case in point, phosphofructokinase. It makes sense what you need to understand. Phosphorylases adds inorganic phosphates, right? They're essentially without the use of ATP. For example, glycogen phosphorylase. So go through these nomenclature, and if you have a good understanding of this said nomenclature, you're gonna have a good understanding of what every enzyme does when they give you these, whether it's in just the glycolysis or gluconeogenesis or any of the pathways, because you have a broad understanding, okay? Now, it's a lot to memorize. I'm pretty sure Dr. Upadhyaya and a bunch of other professors gave you that giant Stanford, I guess, biochemistry pathway sheet. And I looked at that once, had a panic attack, and never looked at it again. So that tells you a lot. What you do need to know is that you should try to whiteboard these pathways out when you guys can. But I'm not going to tell you guys to keep whiteboarding it every single day because you're going to get completely exhausted by by the time you guys roll into your exam time. Okay, so make sure you guys do a proper thorough pass through either whiteboarding or whatever notes that you guys create, and then just review the rate limiting enzymes and the factors associated with them. Because at the end of the day, when you're going through this, you want to make sure you have those rate limiting enzymes down more so than just knowing the entire pathway front and back. Obviously, there are factors that you do need to know very well, such as your glycolysis, TCA, and ETC very well as gluconeogenesis. But every other pathway after this is going to be relatively more focused in on those rate limiting enzymes because the clinical applications start, tailoring off, right? So when you guys cover, your urea cycle, make sure you guys keep that in mind. You don't just sit there and memorize the entire darn thing. You focus in on the high yield clinical correlates, and that's going to make this DM module much more digestible and metabolizable, if that's even a thing. Okay. Glycolysis, okay? Learn this pathway very well, okay? First thing you need to know is PFK1. That one is one of the major regulators of it. And the regulators that regulate the said enzyme is going to be AMP, which is telling you that you're in a low energy state and you need to convert as much of the glucose stores or products that you have floating around into functional ATP, right? Remember, glycolysis only generates at the full yield about like 2, ATP. Remember, the pyruvate that you do generate at the end of the cycle is eventually going to feed into the TCA and have a much higher yield of ATP, okay? Obviously, if you have a whole bunch of energy, right, you have a whole bunch of ATP floating around, that's going to have a negative regulatory function on these said enzymes. And we're going to go over these enzymes and the specific regulatory factors, but keep this in mind. These are what they like to ask about. Okay, they know that you're going to memorize all the enzymes, but they're going to ask you, oh, what happens if a muscle is, contracting a lot? It's using a whole bunch of ATP. What's going to be generated? AMP, right? And what is AMP going to upregulate? Phosphofructokinase 1. Okay, so learn it based off of that kind of narrative, and it's going to make much more sense to you. TCA is very similar in that nature. If you have a whole bunch of ATP, it's like, why do I need to do all this work? So it won't continue the TCA cycle based off of enzymes that are regulated, such as isocitrate dehydrogenase. And NADH is another great example. Remember, NADH is essentially equivalent to ATP, but it generates a lot of, via the electron transport chain, a lot of ATP, right? Because remember, NADH is roughly like 3.2 or like 3.5 ATP that's generated per NADH, okay? Keep that in mind, and you're going to be pretty much solid on these pathways because they love these regulatory enzymes. Okay, this was shamelessly stolen from my friend Brady and Lindsay, but it's a great way to approach this material because at the end of the day, what we're all ultimately talking about is food going into our system or when we're starving, okay? Critical thing, when you are in a fed state, what's going to predominate? Insulin, right? So what you want to do is make sure insulin takes all that wonderful glucose floating through your circulatory system, starts either storing them or breaking them down into energy usable states. How does this happen? It first goes and dephosphorylates a whole bunch of enzymes. And I'll show you guys exactly what those enzymes are for each of the pathways. Glucagon, on the other hand, your body's like, dude, I'm starving. Like, why aren't you giving me energy? Because remember, there are key organs in our body that focuses entirely on glucose usage, right? Like it needs a constant supply. A great example is the brain. It only runs off of glucose. Okay, maybe some ketones for the time being to kind of, tidy you over, but it's constantly need glucose. But where does most of your body's glucose stores hang out? Liver, somewhat in the muscles, as well as you can do a little bit of recuperation of your glucose stores from the kidneys as well. You guys will remember that from your CPR days. Glucagon is going to predominate and help you with capturing those glucose and getting it to the targeted organs that are eventually going to use them. Remember, you want to take it from the stores and make it available for the rest of the body and the organs to use, okay? Remember, increases blood glucose, makes glucose available, and then breaks down glycogen. That's the key kind of principle to keep in mind, and it's phosphorylation via kinases, okay? Phosphatases, kinases. Phosphatases dephosphorylate. Kinases are going to help with the phosphorylation, okay? Let's go through some glycolysis, okay? Remember, glycolysis is not the biggest thing. It's not the biggest kahuna of the whole kind of family of biochemical pathway. But what you do need to know is that these rate limiting enzymes, as well as factors that require ATP. Remember, if you guys think back to your days of FTM, why the heck do we take glucose, which is a beautiful product by itself, and we add a phosphate group to it? It serves two functions, right? Remember, glucose and any sort of other particle that's floating around in your system is osmotically active, right? Wherever you have high stores of a solute, water is going to try to come in. So when you guys think of it this way, when you add that phosphate group, you make water not play a role in having the cellular kind of exchange, right, of water from extracellular space to intracellular space, as well as keeping glucose and fructose stored in the cell so that they can't leave. When they're phosphated and they have a phosphate group on them, they're basically trapped inside this kind of jail of a cell, okay? That allows them to retain their function there, and from there, they can go on to be converted into ATP that they eventually, your body will use, as well as your cells, to create, functioning living human beings. Okay, key thing, glucose 6-phosphate is going to be, glucose 6-phosphate is going to be added in via hexokinase, right? And glucokinase. Fructose 6-phosphate is also with glucokinase. But the key thing is you need to know the distinguishing factor between hexokinase and glucokinase and which organs or how biochemically are going to be favored. And we're going to go through that in a bit. Now, fructose 6-phosphate then gets converted into fructose 1,6-bisphosphate, very critical to remember, and the regulatory components that play a role in this enzyme's function, phosphofructokinase 1. Remember, it's the first rate limiting enzyme. You got to know this one very well because there are multiple factors that play with it. AMP, your body's already used up a whole bunch of ATP. Boom, it's going to upregulate phosphofructokinase. Fructose 2,6-bisphosphate is one of the intermediaries that are created in the side pathway that's going to upregulate the action of phosphofructokinase. ATP is going to have a negative feedback as well as citrate, because remember, ATP is coming from the very end product and citrate is coming from your TCA cycle. So think about multiple pathways converging and regulating glycolysis aside from just knowing the pathway from top to bottom. Okay, we've covered all the stuff that requires ATP usage. Now let's talk about ones that, give ATP. Remember, the ones that are yielding actually energy that you can use. One is very critical, phosphoglycerate kinase. It takes 1,3-BPG and it's converts it into 3PG. That's one. And then the phosphoenol pyruvate as well as conversion into pyruvate, right? That's a one-way pathway. Pyruvate kinase, very critical. They love asking questions about this. So if there's any ones that you need to know for sure, hexokinase, glucokinase, phosphofructokinase, as well as pyruvate kinase have to be down solid, as well as the regulatory components on the side. And if you guys run through these and you guys explain it out like the way I do often with group studying, it's going to make so much more sense because you will add a narrative to each of the states that you're talking about here, right? ATP is high, you have a whole bunch of energy. Alanine, your muscles are being well supplied and they have a whole bunch of amino acid alanine, and it's telling you, you don't need to do this. We need to actually create glucose. So alanine is part of one of the things that you're going to cover later on this week. Glucagon makes sense because remember, we want to create glucose rather than break down, right? Glycolysis is the breakdown of glucose, whereas when you need to release it, you want to inhibit this pathway so you can do gluconeogenesis, okay? Okay. Gluconeogenesis. These are the irreversible ones. If you have these down solid, you will not have to worry about any of the other kind of intermediaries because they all are part of glycolysis and the bifunctional aspect of those enzymes, right? They can go either way, creating from substrate to product or product to substrate, okay? Pyruvate carboxylase, need to know it very well. It's coming from the mitochondria. So they love asking these very it feels like first order questions, but they need to ask these questions because they are critical in telling you what is happening at the cellular level, okay? Phosphophenol pyruvate carboxykinase. I love how big these enzymes names get. In cytoplasm, right, it's critical that you guys know this. It's oxaloacetate to PEP, okay? This is, they love asking about PEP. If that's one thing you guys need to know about gluconeogenesis, PEP is the kind of the starting point at this point, okay? Know that very well, and you guys are going to be good. Oh, you guys will also cover glucose 6-phosphatase because that's the, I guess, the last step that you will essentially hit before you make functioning glucose that can be pushed out into the bloodstream, okay? Now, this is very busy, and it's a kind of a pathway that often throws people for a loop. Don't worry, we're going to try to keep it very simple by taking it into two parts, okay? First, let's talk about when you are in a fed state, right? Think about dephosphorylated, insulin is predominating. So fed state, insulin is high, right? That means you're decreasing cyclic AMP, protein kinase A, FBA's activity goes down. So what is that telling you? FBPase2 is one of the aspects of PFK2, right? PFK2 has two different components. Like it's essentially that enzyme serves like dual roles. Okay. FBase2 is in charge of breaking down when you are in a glucose deficient state. So you want to do gluconeogenesis. So here it's actually downregulated so that PFK2 can be more active and create the upregulating product, which is fructose 2,6-bisphosphate. That's going to say, hey, dude, PFK1, why don't you go ahead and start, cranking out all of the wonderful fructose 6 phosphate that I have here and create it into fructose 1,6-bisphosphate. Okay. Once you have this is one of the major steps, right? I told you guys, if one of the important enzymes to know is PFK, PFK2 and PFK1, they love to ask those questions because they essentially would just say, PFK2, you are in a fed state, insulin is high, what's going to happen to PFK1? So they really like to play around with this. And there's a great mnemonic, Prince Frederick, right? Works only when fed, right? Prince Frederick PFK only works when fed, so insulin is predominating. Now, let's talk about when you are in a fasting state, right? Think phosphorylated. What's happening? Glucagon is high. Glucagon is going to go ahead and phosphorylate everything. Cortisol will also upregulate this, so keep that in mind. It's not as emphasized in this lecture, but it is something to keep in mind that glucagon and cortisol are going to work in very similar aspects to a lot of different enzymes. Okay, cyclic AMP goes up, protein kinase A goes up, FBase gets really upregulated. So it takes all this fructose 2,6 bisphosphate that you've created, and it's going to say, let's go ahead and go backwards and push it towards gluconeogenesis, because remember, fructose 6 phosphate can then be converted back into, because remember, we're just essentially tweaking around the molecular shape of sugars in order to make them into glucose. So less glycolysis and more gluconeogenesis happens. A nice way to remember this is Fabian the peasant has to work harder when starving, right? Think about all those peasants out there working oh so hard. This is nicely summarized here. Insulin, right? High blood glucose levels, dephosphorylated state, active PFK2, increases fructose 2,6-bisphosphate, and then allosterically activates PFK1. So think about this pathway, because remember, PFK2 is the primary regulatory enzyme. Everything else is going to cascade down to PFK1, which is going to allow for you to create eventually pyruvate. Okay. Whereas glucagon is going to tell you, dude, we need glucose. Like, please phosphorylate everything. FBase2 gets active and it starts decreasing the fructose 2,6-bisphosphate, right, which is one of the allosteric regulators. So if there's less of it, it's not going to go ahead and activate PFK1. So what is it going to do instead? It's going to take more products that are created of fructose 1,6-bisphosphate and then move it towards a glucose producing or gluconeogenesis pathway. Here's a nice summary so you guys can view it from top to bottom and see how it works. Remember, all of these are going to be working via almost essentially your G proteins. So if you have that G protein concept down from term 1, you're going to be completely fine. Okay. Here is they love asking this. I don't know why. They're just so hell-bent on ATP and the products that are made and lost. So remember, the net yield at the end of the day is only going to be 2 ATP, right? So it's not that much because you've already lost 2 here. And your NADH is essentially useless unless it's brought into the electron transport chain. So if they ever ask you that, net of glycolysis, 2, right? 2 ATPs are made because you've given 2 you've only generated X number of NADH, which is just 2, and it can't really do anything unless it enters the pathway. This is another concept. I'm pretty sure you guys went over this in small group, but higher Km, okay? Don't think of it as like the higher, like higher binding affinity. It's not. Higher Km is actually lower binding affinity, and it specifically makes sense because glucokinase, if you guys think about it this way, is a enzyme. It's a isoform of hexokinase that essentially lives only in the liver, right? Remember, we want our glucose that is, that we derive from the foods and things that we have be brought into the muscles and tissues that need it first before we start storing them, because the liver at the end of the day is a big hogger of glucose. It's going to hold on to the glucose that you make and store it as compared to use it, because your tissues and cells everywhere else are going to need it first. That's why hexokinase is going to predominate in other cells. So they're going to have low Km, okay? Stick with me. Low Km meaning higher binding affinity, right? So they get the first pass at all that wonderful, yummy glucose floating around. But what you have to remember is they have a very low Vmax, right? There's a point of saturation where you hit it, and then glucokinase is like, I got you. I can take care of all that extra glucose floating around. So this is why the way our body works is regulating these enzymes very tightly, so that if you have excess of glucose, the affinity goes up for it, right? Because you have high amounts of the substrate, and that eventually can then be stored by your liver for long-term usage. Another key thing, hexokinase is independent, okay? They like to ask these flipped questions. Hexokinase is independent of insulin, whereas glucokinase is insulin dependent, okay? So if you remember that, it makes sense as to why your muscles and the rest of the tissues in your body get it first, right? The glucose that's floating around in your system versus your liver, which is the last pass who gets it. Mutations are hyperglycemia and MODY type 2. We're going to cover those in a bit. Glucose sensors in the beta cells basically control this. Remember, there are a couple of ways they can ask these questions. Insulinomas, if you have high levels of insulin, like where does all this insulin go? What is the blood glucose value going to be? So they like to ask those questions. Don't get tripped up. Once you guys know the fundamentals, you will be good. We're going to take a little bit of a break and go over the pentose phosphate pathway. I'm kidding. I'm not ever going to teach you guys this pentose phosphate pathway, because you only need to know a couple of critical components of it. It's also a very minor pathway that you guys covered in CPR 1, if I'm not mistaken. What you do need to know is that it happens in the cytosol. I don't know why. They just like asking these cytosol versus mitochondria versus what's happening in the adipose tissue type of questions, okay? Just know that it happens in the cytosol, right? And the glucose 6 phosphate dehydrogenase is the key enzyme. Guys, remember the G6PD deficiency, fava beans, don't eat them if you have like this weird genetic kind of factor, like all that jazz, okay? You don't need to remember all of those again, but you do need to know the enzymes involved in this. Remember, this does not generate any ATP in this pathway, right? All it generates is NADPH. Now, think about it this way. I like to make this fun, right? NADPH is also upregulated when you drink. I'm not talking about like water. I'm talking about alcohol. So NADPH is in higher levels. So that's why you have greater fatty acid biosynthesis, AKA you have a lot more deposition of fats, right? AKA the beer belly and all sorts of other things that, are commonly floating around. There's upregulation of cholesterol biosynthesis, steroid hormone synthesis in the liver. These are all factors that especially, if you guys cover, chronic alcoholism later on, you're going to see that it's a major factor as to why alcohol is so darn bad. Not saying that, one way or another, what you guys want to do with your own free time, but all I'm saying is it's critical. Now, the other component that they like to ask is of the transketolase. Why? You're like, why do I need to know transketolase? It's because vitamins. They like to ask vitamins, and there's a whole series of lectures that are just dedicated to different vitamins and the pathways that they deal with. I don't know if you guys have had that yet. It's probably coming up, but if they ever ask you about transketolase being downregulated or not functioning, it's because of vitamin B1 or TPP. Thiamine deficiency is very bad, okay? So low transketolase activity, it's very, very buzzword heavy. So transketolase, think TPP, or they like to ask B1. Okay, we've covered glycolysis and a little bit of gluconeogenesis. So what happens with glucose? You've created this wonderful, pathway. What is the end product? Well, the end product isn't actually an end product at all. It's just pyruvate, right? Pyruvate from there can go into multiple different things, right? Pyruvate, if there's too much of it, right? And it can't, if there's not sufficient levels of aerobic respiration, it has to go and get converted into, gets converted into lactate, but lactate is essentially useless for us, right? As human beings, because the only place that can properly handle the change of pyruvate to an active form is in the liver. So that's where the Cori cycle happens. So you take all this wonderful pyruvate, you convert it into lactate at the tissue level, you take this lactate, you send it off to the liver, and the liver is like, okay, I'll take all this wonderful lactate you're giving me and I'll convert it into pyruvate and send it back to you so that the tissues can continue. So this is what happens with long distance runners, right? They are sufficiently breathing, but it eventually needs to get converted back into pyruvate so that they can keep going. Remember, there's a big measurement of lactic acid levels in runners in order to make sure that they're optimizing their run, that they can go a longer distance. So there are multiple measures of this, and that's why they spend so much time kind of covering pyruvate to lactate versus lactate to pyruvate, and then the Cori cycle. You can also do glucogenic amino acids, and this is amino acid metabolism that's going to come up to you guys this week. So don't trip on it. We'll just give you guys a broad overview when we're done with this kind of review session. Now, another thing is alanine. This alanine kind of cycle, glucose alanine cycle, is a big component of how you shuttle around a lot of your amino acids, as well as your nitrogenous waste products. And we're going to cover this in a bit in another review session, but I want to pinpoint it out for you guys because remember, ALT. You guys remember what ALT is? Alanine transamylase, like it's one of those enzymes, ALT is transferase. ALT is one of the products that we use to measure liver function enzymes. Do you guys see why we're learning it now? ALT, right? Versus AST. You guys remember a shot of tequila, right? All of those values, we're using them because the liver is a great enzymatic kind of treasure store, and it tells you how the liver is functioning if you are potentially looking at someone with chronic alcohol use, right? What are they going to have? An increased levels of AST to ALT ratio, telling you that the liver is damaged based off of alcohol versus ALT. Okay, ALT, if it's elevated, it's telling you some kind of problem with one, either the gallbladder, or two, that you're having some kind of viral infection affecting it, or you can also have cirrhosis in a different way. That's definitely pathology. You guys don't need to know that. The next component is the conversion. You guys will love this, into acetyl-CoA, because acetyl-CoA is then going to go into your eventually into your TCA cycle and then create your wonderful products that are going to be fed into the ETC, and then you generate out a lovely 32 ATP per cycle. Okay. And then, of course, you can go directly from pyruvate back to gluconeogenesis. So what you do is create an intermediary kind of pathway of creating oxaloacetate, and from there, oxaloacetate can then go into gluconeogenesis. Okay. Here it is, the Cori cycle. Keep this in mind. Cori cycle is very critical. It happens at the RBC skeletal muscle level, right? But the 2-lactate that you generate from the 2-pyruvate, it's telling you that the body essentially can't use this unless it gets passed on to the liver, and from the liver it gets made into glucose, right, via almost a gluconeogenesis pathway, and then it gets brought back into the circulation and eventually used up. Remember, we can only do so much of the Cori cycle before we have a lactic acid buildup, and that's why your muscles hurt after a really long workout or a really nice run. Okay, keep that in mind. All right, we get to the lovely TCA cycle. Everyone loves this. Pyruvate dehydrogenase, okay? Say it after me. Tender loving care for Nancy, okay? What does this mean? You need to know all of these factors for pyruvate dehydrogenase complex because they love asking these questions and they're applicable for all of these enzymes, okay? PDH, alpha ketoglutarate, and then branched chain keto acid dehydrogenase. They all use tender loving care. So if you had to take a hedge, right, hedge a bet, right, that if there's a dehydrogenase, most of the time they're going to need tender loving care for Nancy, which is thiamine, right, which is the B1. What is another thing that's messed up? If there's a thiamine deficiency, TPP, transketolase. Look, guys, you got to make those connections because they will ask you about multiple pathways back to back, and they won't say like, tell me what goes wrong in your pentose phosphate pathway. They're just going to say, patient presents with thiamine deficiency, has issues. You can tell that their pyruvate dehydrogenase is downregulated. What else? What other enzyme do you think is going to be downregulated? And you have to make that connection that transketolase is dependent on thiamine, right? Thiamine pyrophosphate or B1. Lipoic acid, CoA, and then FAD and NADH are all ones to keep in mind and memorize because they love asking any versions of these names, okay? There are a couple of mnemonics floating around. Feel free to drop it in the chat. Just keep it rock solid. And the dirtier the mnemonic, the better it is to keep it straight. Okay, pyruvate is oxidized into acetyl-CoA, right? And eventually goes into the TCA cycle, right? But pyruvate dehydrogenase is a complex. Whenever they say the word complex, it's telling you that that enzyme isn't just one enzyme. It is a series of different enzymes or proteins, essentially, working in tandem to do essentially a multitude of different functions. Remember, pyruvate dehydrogenase is tightly regulated because it's an irreversible enzyme reaction. So your body has to be very careful and diligent when dealing with it. So if you have excess of acetyl-CoA, it's going to downregulate it. And if you have excess of NADH, it's going to downregulate it. What does this tell you? It tells you that acetyl-CoA, which we can generate in multiple pathways, yes, alpha ketoglutarate is also affected in this. If you guys are messaging me, I'm not keeping an eye on the chat, so please message it to Priya or put it in generally to everyone so that everyone can benefit from any sort of questions being asked. Just want to throw that out there. Pyruvate dehydrogenase complex is composed of pyruvate decarboxylase, which is E1, dihydroxypolytransacetylase, E2. I swear I'm going to butcher all these names. Dihydroxypolyl dehydrogenase is E3. You really don't need to know those to that level of depth. All you do need to know are the regulators, right? Think about it this way: acetyl-CoA and NADH are the primary regulators, but there are others that you should keep in mind. What are the other ones that deal with pyruvate dehydrogenase, guys? Throw it in the chat, but I'm going to give you guys a couple of them. What are they? First, let me ask you guys that before I... PDH kinase, okay. What's going to upregulate it? What's going to upregulate pyruvate dehydrogenase? AMP, insulin. I love that. Okay, make sure you guys make those connections because this is not an isolated thing. It's not just like you need to know like the products that are being made because they will go ahead and ask you those multi-layered questions of fed versus fasting state. Okay, cool. Let's look at a couple of things. Here it is. You guys are already mentioning it. Calcium, right? You have a whole bunch of calcium. What does calcium do? It is involved with muscle contraction. So calcium is one of the things that's going to upregulate your PDH, right? Makes sense because your body's like, dude, give me more energy. And you're like, sure, go ahead. Pyruvate dehydrogenase, like, I got you. Cardiac muscles will release catecholamines. And you guys already said this. Catecholamines, right, are going to do a similar function to glucagon, increase your blood glucose levels, right? This is why like stress and things make you feel like you're alive. You're like, because your body's like, dude, you're in like, you're going to go fight a bear right now, which is essentially an SGU exam. It's going to upregulate, push a whole bunch of glucose into your circulation and help you get it to those target tissues and organs to then keep you awake and alive for the exam. Important regulators are NADH, acetyl-CoA, pyruvate, ATP, and calcium. Okay, if you have that down, you should be solid. Know that its high energy charge is going to turn it off, right? Whereas low energy charge is going to upregulate it. Makes sense, right? And let's cover activation, right? Activation is by phosphatase, right? Because remember, by dephosphorylation of serine residues on the E1 PDH, the PDH phosphatase then can go, the PDH complex can then go and start converting more of the pyruvate into acetyl-CoA, okay? Phosphatase is stimulated by 1, calcium, 2, by insulin, right, from the adipocytes as well as in the liver, catecholamines for the cardiac muscles. Inhibiting factors, okay, if you have for the E1 complex, right, PDH kinase, it's going to be inhibited by pyruvate, right? If kinase is inhibited, then the complex is not inhibited, right? The kinase just says, okay, look, if I have a whole bunch of pyruvate, the pyruvate saying, I'm going to go bind to this slot in the E1 state and tells them to go and upregulate the PDH because there's a whole bunch of pyruvate. Kinase gets activated by ATP. ATP meaning you have a high energy state. You have high levels of acetyl-CoA already. It's a backup. And NADH is also another backup because it's all products that are generated via a dehydrogenase process, AKA it will shut down the complex. Once again, tender loving care for Nancy, cofactors of NADH and coenzyme A, which is B5. Keep that straight. There are a lot of B enzymes. They will cover an entire lecture of all the enzymes with you guys, okay? This is very sad. Pyruvate dehydrogenase deficiency, it's not as common, but it is one that has a lot of very clinically significant presentation, frontal prominence, wide nasal bridging, flare nares, long kind of difficulty, one, brain development problems, as well as corpus callosum agenesis, right? As well as cerebral and basal ganglia, which are not good. You guys are going to cover a little bit of this in NB, okay? Thiamine deficiency, you have to know. What is thiamine, guys? Thiamine pyrophosphate. What enzyme, what vitamin is that? Put it in the chat. There you go, B1, okay? They love asking those multitude of ways. Know those B enzymes, right? The numbering for all of them as well, because it's in the subsequent lectures that you guys are going to cover, and it's high yield. Wernicke-Korsakoff is one that is caused by nutritional deficiency, and it can also be caused by alcoholics. It's just one of those things that goes on with this pathway, because remember, the pentose phosphate pathway gets upregulated, right? You're generating a whole bunch of NADPH. Your body's like, I'm technically in the fed state, so I'm gonna start storing all the fats and proteins and everything that I have, which leads to a lot of presentations that you see with alcoholics, right? Especially chronic alcoholics. Ataxia, ophthalmoplegia, memory loss, cerebral hemorrhaging, confabulations. Those are all buzzwords, okay? If you know these, okay, you will know right off the bat Wernicke-Korsakoff. Less so that is often covered for you guys, because this is going to come up more in like upper, subsequent terms. It's the berries, wet versus dry. Wet beriberi, just think about dealing with vasculature, right? Heart failure, decreased ATP, increased cardiac output. It can present with cardiomyopathies. Whereas dry beriberi is just systemic muscle wasting. It sounds kind of weird, but if you guys are passionate about working out, alcohol isn't the way to go, especially if you have a drink right after you work out. It actually has counter anabolic effects to the muscle building. And if you have it for long term, such as with thiamine deficiency and chronic alcoholism, it's going to lead to a whole bunch of muscle wasting, which is what you see if you've seen a long-term alcoholics. Their like sides are very much atrophied, their arms and legs are very much atrophied, and they have a lot of fat deposition centrally. Other thiamine required, right? Alpha keto glutarate dehydrogenase, which is part of the TCA, and then the branch chain keto acid dehydrogenase, which you guys are going to cover later on. Okay. Look, this one, they love asking this question. Heavy metal poisoning, as well as with arsenic and arsenate or any sort of things, it's going to affect two specific components, right? What is one of the enzyme that gets messed up by arsenic in your glycolysis? Throw it in the chat. There you go, G3P dehydrogenase, which is part of your glycolysis, right? The other thing that gets messed up is from your TCA cycle. Believe it or not, pyruvate dehydrogenase with arsenic or mercury or even lead poisoning gets shut down as well, right? Plus, a deficiency in lipoic acid can also result in the pyruvate dehydrogenase being arrested, okay? Multiple enzymes are affected. They like to ask these arsenic level questions for all these enzymes and tell you like which cycle will be downregulated, which enzyme is going to be downregulated. So keep that in mind and you guys are going to do great. TCA. We finally get to TCA, okay? You've taken all that effort to create pyruvate. From pyruvate, you're going to go to acetyl-CoA, okay? Acetyl-CoA to citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. You guys have to know this entire pathway. I am so sorry to tell you guys that, but you have to know the entire pathway as well as the irreversible enzymes. If you have these two pathways down, you're pretty darn solid already, okay? Now, also know the cofactors, right? Vitamins B1, B2, B3, B5, and then lipoic acid are all things that they like to test on. They will ask you guys questions about this, I guarantee it, because they asked us this multitude of different ways, okay? The end goal of the TCA cycle is to create high levels of one, NADH, right? One level of FADH, generate, a little bit of carbon dioxide, as well as, take all this net yield of GTP and then produce about 10 ATP per acetyl-CoA. So you get about 20 acetyl-CoA through the entire TCA cycle. So it's a big chunk of energy that you get on the regular basis. So TCA, remember, it's an aerobic respiration. And remember, where is it happening? It's happening in the mitochondria, okay? If you remember that, you're going to be good. Once again, the highly tested kind of components is going to be citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase, okay? All of these are going to be tested, and they like to ask those based off of whether it's the product that's being created that's eventually going to have a negative feedback, or if there's other kind of components that are going to upregulate. For example, with isocitrate dehydrogenase, it's the same kind of conversation we previously had. High levels of ATP, high levels of NADH, which are all products of the TCA cycle. It's going to say, don't kind of make more products than we need. If you have low levels of, let's say, sorry, not low levels, but if you have low activity of isocitrate and your body's in a very energy deficient state, you're going to have what? Excess levels of ADP, right? And then you're going to have high levels of calcium, which is telling you what? It's telling you that your muscles are using up energy, right? And that you're creating less energy favorable states, such as the products that are created with ADP, right? Or AMP. These are all products that are used to tell your body, hey, you're in an energy deficient state. Let's upregulate these enzymes so that we can create the products that we need to get this ball rolling, okay? Your alpha-ketoglutarate dehydrogenase, it's very similar to pyruvate dehydrogenase because it's a complex, right? So it has multiple components, E1, E2, E3. Don't need to really worry about it, but what you do need to know is those coenzymes, right? Tender loving care for Nancy, if it's a dehydrogenase, good chunk of the time, if you guess, tender loving care for Nancy, you're going to be fine. What about if it's activated by calcium? Why does that make sense? That's because your body's like, hey, I'm using up all this calcium. You might as well upregulate the enzymes that are here in the mitochondria to make sure that you're pushing this cycle forward. Obviously, it's going to be inhibited by NADH and succinyl-CoA. And succinyl-CoA, remember, is a product that you created from the TCA cycle. Malfunctions, okay? Fluoroacetate inhibits acanatase, because remember, acanatase is one of those things that you need to keep in mind. It's one of the major rate limiting things, right? It goes from citrate to isocitrate. They like to ask about acanatase because it's one of the less things that students focus on. And it's basically going to lead you to an excess levels of citrate, just hanging out. And we can talk about what happens to that citrate when it eventually, gets converted into something else. Malonate inhibits succinyl dehydrogenase. So remember, it's a competitive inhibition. So they like to ask those questions, as well as any sort of vitamin deficiency. Remember, those are coming up in your lectures, as well as cobalamin. Okay. They like B12 for some reason. Remember, any sort of B12 deficiency, it's going to present with macrocytic anemia. So if you see macrocytic anemia, start thinking B12, what enzymes are going to be shut down thanks to this vitamin deficiency. So what's the yield? What are we getting after all this effort? 1 TCA round gets 3 NADH, which is about 9 ATP. 1 GTP is equivalent to 1 ATP. Remember, you're going from guanine to adenine, so it's not a big deal. 1 FADH creates roughly about 2 ATPs. So overall, 12 ATP per acetyl-CoA. So remember, you have 2 acetyl-CoAs, so it's like roughly 24 ballpark range. And you're going to do a little bit more with your electron transport chain, which is what we're going to cover next. Guess what? Okay, guess what? You don't need to memorize this entire thing. God bless, right? That's one less thing you do remember. You do need to know that there are 1, 2, 3, 4, 5, 6 components to this entire madness. They really don't spend too much time covering coenzyme Q, but it is important because it's a lipid-based, okay? So if you give any sort of statins, it's going to downregulate or have less enzyme like coenzyme Q, and that's going to be problematic because remember, it's one of the ones that helps carry those electrons from one complex to another. So there isn't too many complexes, and they have nice names for it. There's NADH dehydrogenase, succinate dehydrogenase, cytochrome C reductase, and cytochrome C oxidase, okay? Those are the major ones. But what you do need to know is that complex 5 is the main kahuna of this whole family, because what you're doing is setting up a gradient, right? You're setting up like the energy complex that you need to eventually turn this. If you guys look at the actual picture, it looks like a rotor system. It takes and forcefully combines ADP with one inorganic phosphate and spits out an ATP. If you guys remember that, you guys are good that it's complex 5. You're 5 out of 5 on this entire, pathway, you're going to be good. What you do need to know is the things that inhibit it. I love this because First Aid does a good job of giving you mnemonics for remembering it, because all the complex ones like end in with 1, right? Retinone. Metformin is less covered because it's kind of a statin and things that we will cover with coenzyme Q. But when you guys are going through this, there's all of these that you should commit to memory because they are tested, and they like to ask those. What happens if you give like an insecticide? Which complex is inhibited versus a bacterial antibiotic? Antimycin, you guys have heard before. Cyanide poisoning, it's because it messes with complex 4, right? Cytochrome C, C for cyanide. That's how I remembered it. It's all the ides, right? If you remember the ides, you will remember that it's complex 4, okay? Now, carbon monoxide, right? As well as only weirdness is all heme kind of components with copper. That's the only odd exception, but you should be good if you remember these that are outlined here, okay? Oligomycin, just remember that's the very specific one. It's an antibiotic that we use. I don't know if we use it regularly now, but it inhibits ATP synthase. Okay, that is one of the inhibiting complexes. There are also kind of ones that you can keep in mind in terms of what either sets up a gradient and breaks it down, or there are ones that are just energy releasing. So inhibitors of ATP synthase, right? Essentially, it's not going to have too much of an effect, right? You're just setting up a gradient. Protons will eventually leak back by, kind of facilitated diffusion or uncoupling by therminogen. We're going to talk about therminogen in a second. And then uncoupling activity of ETC via ATP synthase. So remember, like I said, less ATP, and that's going to be problematic. Inhibiting ANT and ADP transport, because remember, ATPs technically has all these inorganic phosphates that have a lot of charges that are being held at different positions. So it's not very easily soluble and passes through any sort of membrane. So what happens is you have your ATP, ADP essentially set up by a pump, right? It's an exchanger. It basically takes one ADP and spits out an ATP. But if you inhibit it, you can't set up that ADP that you need to build up eventually to create those ATPs, right? Via your ETC. Atractyloside, right? Just keep that in mind. It downregulates it, as well as bronchitic acid. This is another one that basically shuts down ATP, ADP kind of transport. So the effects are very similar to oligomycin, but you just aren't setting up a gradient, right? So complex 5 can't dissipate the proton gradients. So just keep that in mind. ETC will eventually shut down, and that can lead to mitochondrial toxicity. Uncoupling of oxidative phosphorylation. Okay, these ones commit to memory. Priya and I, we definitely covered aspirin way too much for our own benefit, but this was something that came up again in term 4. So keep that in mind. These are all cumulative knowledge that we're going to keep building up on. DNP-24-dinoproterenol is a synthetic uncoupler. ASA, aspirin, right? Gramicidin, which is basically forms a pore and just lets out all this hydrogen that you've built up on the intracellular membrane and brings it back into the mitochondrial matrix. And then the vancomycin, okay? And then uncoupling proteins. This one is very critical because we endogenously make it as babies. We have thermogenin. Fun fact, we, back in the 80s, I think they used to give this artificially synthesized version to patients to burn up it was helpful for weight loss and making sure the patients are getting to their target weight and stuff, but it actually had a lot of adverse effects, so we no longer just give this willy-nilly to patients. What happens is it decreases ATP synthesis and increases electron transport chain as well as oxygen consumption, right? Because remember, oxygen is the final carrier in this whole pathway. With the uncouplers, this is the problem. You're going to kind of burn up energy as you're kind of bringing these charges down. Okay, uncoupling via oxidative phosphorylation. This can happen, right? It's released as heat. A biological function that all babies are born with is thermogenin in their brown fat. Remember, brown fat, we no longer have it. Unfortunately, because if we had it in our adipose tissue, we'd all be like, I don't know, abs for days and muscles for days, okay? But it's a biological heat pad for babies because they need to be at a higher temperature. That's why like most of the babies that you often see that are newborns are all like wrapped up, like they're all toasty as heck, whereas like we would like nothing better to like live and die in an AC, okay? ACP1 is a thermogenin, is expressed in brown fat, so keep that in mind. And any sort of ionophore or any sort of oxidative phosphorylators are all going to eventually, 1, increase oxygen consumption, or 2, release heat. So if you hear those buzzwords, start thinking of these and start, making your correct answers available. Okay. This is what I was telling you guys about bronchitic acid, as well as what was the other one that I always forget? Actyloside. They both are essentially going to just downregulate the activity of this adenosine nucleotide translocase in exchange of ATP for ADP. It's an antiporter. If that is downregulated, it has very similar effects to you inhibiting complex 5. Okay. All right. So this is the net yield that I kind of was talking about for the NADH versus FADH, right? It's roughly about 3 ATP versus 2 ATP. So obviously your body will prefer for you to make NADH, but it can basically live off of the stores of FADH, which is generated in other pathways as well. So keep that in mind. Don't even trip. Okay. Mitochondrial deficiencies. Why are you guys covering it here? It's because this has downstream effects on, most of these are pretty darn terminal. And it's unfortunate, but it can lead to a lot of different kind of presentation. I really use Picturize for this to kind of keep these straight. Leber's hereditary optic neuropathy, any sort of like blindness, any sort of maldevelopment, all of these are going to present very similarly, but they're very buzzword heavy. So if you guys remember your MELAS versus your MRFs, right? MELAS has a lot of mitochondrial myopathies, encephalopathies and all of those things. Whereas MRFs, right, it has ragged red fibers. So that's how I kept it straight. MELAS versus MRFs, right? Ragged red fibers in MRF. There's a lot of R's in MRF. So ragged red fibers. and they have a lot of myoclonic seizures. Okay. Cool. This is the entire shebang that we've covered thus far. Let us move to glycogen. Okay. So this is the primary energy kind of storage format. Obviously, our adipocytes have fat stores in them, tags and cholesterols and all sorts of other stuff stored in there. But glycogen is a big darn component of what makes up our liver and everything else around it. Okay. It's primarily stored in the liver and the muscle. If they ever ask you, go with liver first. And if you don't see liver as an option, go with muscle, because that's what they like to ask. Glucose, right, is just basically the subunits that make up this. What they essentially will ask you on is what type of linkage will they have? Alpha 1,4 versus the branching points is alpha 1,6. Now, why do you need to know this? Because in order to create these, the branching and the debranching enzymes, there are disorders with them. Okay. Also very buzzword heavy is glycogen. It's the core, right? It's just a core protein. Don't say that it's a sugar. It's not a sugar. It's a protein that essentially enables for the scaffold that makes up this wonderful glycogen. Okay. And yeah, we're going to cover starches. Okay. They like to ask questions about this. This was less tested, but I wanted to make sure you guys get these points. Remember, starch is different glycosidic linkages, right? It has a linear alpha 1,4, and then branching is alpha 1,6. There are very few foods, right? I mean, there are certain type of foods that are preferable, right? Because they help with peristalsis as well as making sure bowel movements are regular. But what you do have to consider is giving them sufficient amount of waters, because unfortunately, starches are still osmotically active, and water is going to be lost if the patient's already dehydrated. You don't want to give them a bunch of starches and fibers and other stuff. Okay. We have salivary and pancreatic amylases. So you guys probably had Dr. Bandolo already, and he started covering all of these either late this week or you guys had it covered next week. So keep that in mind. They are going to come back. Cellulose, unbranched homopolysaccharides. Okay, glucose is making up the backbone, right? It's dietary fibers. Glucose, right, is the backbone. It's beta 1,4 linkage. It cannot be cleaved by us. This is why we give them plenty of water, because it's osmotically active. Lactase, it only hydrolyzes beta 1,4 linkages that's found in lactose, not in cellulose, okay? So don't get tripped up, beta 1,4, thinking that beta 1,4, if I memorize it, I'll be okay for the exam. It's beta 1,4 for cellulose, for glucose products, because remember, lactose is glucose and galactose, okay? Reducing sugars. Now, sugars, you got to keep these straight because they're actually used clinically. They will test your urine for sugar content and kind of like how much is getting passed out, right? Glyceraldehyde versus dihydroxyacetone. You have a aldehyde group versus a keto group. And you guys are going to cover these. Well, actually, we'll cover it today with your ketones. Lactose, right? What you need to know essentially is beta 1,4. A lot of people don't have this kind of linkage, whether it's hereditary or familial or et cetera. Or if it's an adult onset. Keep this in mind. You do need to know that lactase, right? Brush borders have, essentially, it's happening at the brush border level, and it's not found in the urine, okay? This is very buzzword heavy. If you don't find it, then you start thinking of lactose kind of levels of disorders. Sucrose, okay? This is the yummy stuff that's found in a lot of drinks. So it's glucose plus fructose. Remember, it's a non-reducing sugar, so intestinally it gets converted via sucrase, and it can be broken down, eventually either stored or released. What you need to remember is that it needs to be avoided in hereditary fructose intolerance, because an enzyme is deficient that is no longer being able to take the fructose and phosphorylate it and make it available for the cell to convert it into a glycolysis pathway, and two, it can lead to like cataract developments and other things. So the refined sugar, so keep that in mind. Maltose, they don't like to ask too much about it. It's an alpha 1,4. It's intestinally kind of broken down by maltase, right? Most of us don't have issues with this. It's because it's just 2 glucose unit, and we have alpha 1,4 ability to break it down. Okay, this leads to our glycogen storage disorders. We're going to cover those more in depth in subsequent ones, but I want to cover a little bit of broad overview of your gluconeogenesis. 'Neogenesis. Remember, the goal is to provide blood, cells, as well as your brain, as well as your renals, essentially needing those glucose, right? Because remember, glucose has multiple roles in both energy as well as keeping your cells in the brain alive, because that's the only energy that they will take, as well as your renal medulla needs glucose for osmotic kind of activity of water, as well as retention of said fluids. One, we can also synthesize glucose from non-carbohydrate sources. We can use amino acids. We can even use our tags essentially to kind of work backwards and create this. Remember, it's the sole provider. Once the glycogen stores have been depleted, you're essentially going into starvation state, and you're going to start making ketones. Okay, we're going to go over the ketone pathway. Here's the pathway. This is what we covered: glucose 6-phosphatase, right? Fructose 1,6-bisphosphate, PEP carboxykinase, and pyruvate carboxykinase. Sorry, carboxylase in the mitochondria. I'm getting super tired. I can already tell. But if it's a carboxylase, what cofactor do you guys need? Do you guys remember? Drop it in the chat. TPP, some people are saying TPP, some people are saying biotin. I'll give you guys a nice little mnemonic. Typically with any sort of carboxylase, you guys remember ABC, okay? If you remember ABC, you're gonna be good. What does ABC stand for? There you go, acetyl-CoA, biotin, and carbon dioxide. Those are the key factors. Those are the things that's gonna help keep your carboxylases moving, okay? I wanna make sure you guys emphasize and learn those points, because those are what they like to ask, instead of just purely like pyruvate carboxylase, tell me what it does. It's never a first order question with those. Okay, these are the other pathways you guys are going to cover, right? We can do these for days, but what you do need to know is that glucose alanine cycle is very critical. Any sort of muscle degradation leads to alanine being released, and then the alanine can actually be fed directly into the gluconeogenesis cycle and lead to the production of glucose, which is going to keep your organs, essential organs, well supplied with sugars, right? Which is essentially glucose. Pyruvate is mainly formed from lactate and alanine, and it enters the mitochondria. And you guys will cover beta oxidation and why we would do beta oxidation in times of desperate need. Okay, these pathways I'm going to go over with you guys a little bit more in depth. Alanine amino transferase, what does this guy stand for? ALT, right? One of the liver functional enzymes that you guys use on the daily basis to see if the liver is functioning okay. So there is some practical use to this knowledge. Okay, usage of gluconeogenesis amino acid for gluconeogenesis. Essentially what you need to know is that alanine and glutamate are the primary things that are going to be involved with this pathway. Do not put anything else. If they ask you a question about this, it's alanine and glutamine, okay? I know you'll be tempted to put alpha-ketoglutarate or any sort of the other ones. Don't, okay? Trust me, it's alanine and glutamine. That's the only ones you need to know that are glucogenic amino acids. Okay, tags. Essential to remember: fatty acid degradation. Okay, fat cells are going to degrade tags only at low blood glucose levels. Remember, we're talking about prolonged state of starvation, and they're going to release free fatty acids for energy metabolism. We're going to do beta oxidation with them. It's going to be great. Everyone has a good time except for your liver, because it's doing the primary effort in this entire path. Okay, glucose 6-phosphatase is found in the liver and the kidney cortex. Why do you need to know this? Because these are the primary sites. Whenever they ask you about gluconeogenesis, where it happens, they will often trick you and not put liver on there, and you have to pick kidney, right? Which makes no sense to you guys at that time, but it is one of the locations where glucose 6-phosphatase is involved with recapturing any sort of phosphorylated sugars, such as glucose or fructose, at the kidney level, and it's going to take that glucose and dephosphorylate it and put it out into the circulation so that your body can use it up again. Okay. Here's the pathway again. I don't want to beat a dead horse with this. I gave you guys the entire pathway that you guys can whiteboard. Feel free to use this. It's color coordinated, pulled directly from AMBOSS, so it's decently high yield. And what I did during my time was I took this pathway and then I added every single factor that plays with this, right? For here, I put like liver and kidney, and then I would then go specifically and then say like, okay, for pyruvate carboxylase, what factors do I need? I need A, B, C. So I would just run through it that way. That was my initial pass of the material. And then eventually I got so proficient that I could just spit out random facts like this. Okay, embryology. God bless for having to leave a little bit of biochem behind. Okay, now, most of your system is broken down into foregut, midgut, and hindgut. If you guys can keep these straight, as well as the divisional lines for them, it's going to be critical, okay? Remember, the foregut starts at the pharynx and ends up at the duodenal major papillae, okay? Critical, major papillae, everything above, foregut, everything below, midgut, and then if you go really far down, it's hindgut. But like, we never care about the hindgut at this point. Duodenal distal to the major papillae, right? What are we talking about the major papillae, right? That's the second part of the duodenum, right? It's where it's like the draining point of the bile duct as well as the pancreatic duct. And essentially anything above that line is foregut, anything below is midgut, okay? And it stops at the 2/3 transverse colon, okay, guys? Make sure you guys highlight these because they like to ask the end as well as the starting point for all of these kind of derivatives. It's very highly tested and then they like those questions. Derivative of the hindgut, right? It starts at the 1/3 transverse colon and goes all the way down, eventually ends in the anorectal canal. Okay. Cool. Okay, a little bit of the autonomics. I don't want to spend too much time because you guys know a lot of this and you guys have like learned this time and time again from term 1 all the way down to like, you know, even in term 2 with ER and a little bit with your DM module. Okay, general layout, right? Your esophagus, stomach, liver, gallbladder, pancreas, spleen, your first half of the duodenum, right? Especially above the major papillae, it's all celiac, okay? Don't get tripped up if they ask you about the duodenum and they say 1/2, you're going to be tempted to put something with the midgut. Don't. If it says 1/2 above the duodenum, celiac, okay? Still blood supply. Superior mesenteric, right, is going to be mostly involved with the duodenal distal to the major papillae and all the way down to the 2/3 of the transverse colon. And then you're going to cover, you know, the last portion of the transverse colon all the way up to the anorectal junction, which you're going to get to the pectinate line and all that jazz. Okay, sympathetic components, right? For the sympathetic components, it's primarily done through the ganglia, right? It's the greater splanchnics, midgut is the lesser least splanchnics, and then finally lumbar splanchnics for your sympathetics. Okay, this entire thing, they do ask about dermatomal level, so T5 to T9, T10 to 11, 12, okay. So those you got to know, and I think there's a nice table that I put together, or I mean, that was put together in your lecture that I threw in here. Okay, visceral pain. Guys, when you're talking about visceral pain, is it going to be sharp or is it going to be dull? Dull, okay, fantastic. This you have to know, okay? Remember, they like to ask these questions and it feels low yield, but it is those points you can capture very easily. Anything at the pelvic pain line and below, right, is travel with the, pelvic pain line and sympathetics, right, until the midpoint of the sigmoid colon, it's going to follow parasympathetics, right? Everything after that, you got to keep that in mind in terms of the pudendal. Okay, sympathetic versus parasympathetic is critical in this exam, and it's a continuation of your ER knowledge. Referred pain, I swear to you guys, I thought they wouldn't ask us these questions, but they asked us in terms of dermatomes, right? Like, let's say what happens with the liver, right? Like, which dermatome is it going to present in? And I was sitting there wondering, where is T5? Okay. So keep that in mind. They like to ask these based off of referred pain. Any sort of foregut structure, T5 to T9, any sort of midgut, T10 to 12, and then your sympathetics is going to be L1, L2, and then finally any sort of below pelvic pain line, right? It's going to be S2, S4. Okay, it's via the pudendal perineal kind of pain pathway. Gallbladder, they love this. Gallbladder versus appendicitis. Okay, know the pain points. Okay, now if it's going to be generalized pain, it's going to happen at the shoulder level because most of the time with any sort of pain like this, it's going to ascend via your it's very similar to chest pain. Don't get tripped up. If you have pain on the right-hand side, on the shoulder level, they're talking about a gallbladder. It's not pain with your heart. Do not put heart. That was a question on our exam, okay? The appendix, right? If there's any sort of pain that's generalized, right? Visceral pain, it's going to be localized to the tummy, belly button area of T10, okay? When the pain gets so bad that it's sharp and localized, that's when you're likely going to rupture the appendix, and that's pretty bad. So if the patient is saying like, oh, my pain is like located in the right lower quadrant, you should be starting to get really worried because it's happening. It's a very sharp localized pain, and that's telling you it's going to be somatic pain versus visceral, which is more localized. Okay. All right, so this is the table. Memorize the heck out of this, as well as the boundaries, right? We're talking about the esophagus as well as the duodenum. I'm keep on emphasizing this because a lot of people were like, oh yeah, I remember it. And then like week 3 of DM, they were like, I have no idea what I covered for week 2 and 1. Okay, embryology, let's continue with this. Remember, your entire body is symmetrical when you're developing. And that's a bit of a struggle because everything's happening all at the same time, but our internal organs are basically solid structures until they're not. So they have to be turned into a hollow tube, and then they have to kind of twist and turn until they properly are nestled into the state that we know anatomically is correct for even a baby, right? They start off as a hollow tube, right? Because remember, we all look like little worms inside of our developing mother's womb, okay? When we eventually get to the point where we have to canalize, right, which is basically, recanalize, we first have to go in and have these epithelias, one, apoptose to a certain degree, and then finally allow for them to properly create the lumen or the cavity that you would need for food and things to kind of pass through. So that recanalization process is essential. When it, one, either properly doesn't happen, it causes stenosis, right? It basically is improper canalization. So it's a tighter luminal space. Or you can have an atresia, which is straight up a blockage, which is bad, okay? And if you have any sort of these, it can lead to a multitude of different presentations. And it's something they like to ask, okay? This one is relatively simple, okay? Anything anterior, right? Organ structure wise, right? Because remember, I told you guys, everything in our body is symmetrical until it's not. Anything that's anterior to the front, such as like the aorta or anything like that, it starts shifting. It starts moving to a place where it's more better situated, right? So it's going to move to the right. Anything that's located posteriorly is going to move to the left, right? So if you guys keep that in mind, it makes sense why things shift the way that they do, okay? Now, let's actually get to the clinical correlates that they like to test on. Congenital hypertrophic pyloric stenosis. Asking the very critical question is this going to be bile stained or non-bile stained? Non-bile stained. Fantastic. Okay. That's because this is happening at the pyloric junction, right? It's between the point where the stomach gets turned into the duodenal component, right? When you have a stenosis of this area, when you're throwing up, there's no bile because you never gotten to the duodenal papillae and the draining point of where bile actually enters the intestinal system. Okay, so keep that in mind. It's a hypertrophy of the muscles. It's a stenosis, so it's not a proper canalization. So they can ask those questions in a multitude of different ways. Okay, here it is, non-bile staining. We got an example of this image on our exam. So projectile vomiting, non-bile staining. I don't know. I just imagine a baby just like spit firing like all this vomit, which is kind of sad, but at the same time, a wild image to have in your head. Now, buzzword for this is pyloric mass, olive shaped, right? If you're palpating the baby and you feel kind of like this like little notch or anything, that's telling you a indication of a pyloric stenosis. And other buzzword is visible peristaltic waves. Okay, if you guys see visible peristaltic waves, do not put Hirschsprung disease if it has in the question stem an olive-like mass, okay? People in our term got tripped up by that and they missed a couple of points, okay? In the same image, you guys can run through your radiograph images. Make sure you guys identify all the parts because they do like to ask about those. And it's very first order questions that you guys can get right without, tripping too much, okay? Development of the duodenum. Remember, the duodenum is up to the, the mid, I think the 0.2 area of the duodenum, everything is foregut. Everything after that is midgut. The other thing that they like to ask is the transverse component. What passes above it? It's the SMA. Because remember, your vasculature at this point via the descending aorta is like right here. Oops, let me go back. I don't know why I did that. It's like just descending normally, right? But here, when you get to a portion of the descending aorta, the SMA, right, and the venous component, right, are all going to be popping outside, right, outside of the duodenum. So the duodenum is actually more posterior, especially the transverse duodenum is more posterior to the SMA. So if there's any sort of stenosis or any sort of food or anything obstructing in that area caused by stenosis, what's going to be likely affected? SMA. So that was a question that we had as well. Okay, this one is a good one. A dilated duodenum, okay, you're going to see above the site of stenosis. That's because food can't pass through nice and clean. You can also have an atresia, which is just a straight up like nothing, right? There's no canalization there. It's basically one solid giant mass. And if you guys remember, a duodenal atresia is caused by polyhydramnios. Polyhydramnios meaning excess amniotic fluid. And this is something you guys covered in term 1 versus, a duodenal stenosis, which is going to be bile stained because this is after, you've crossed your duodenal papillae. Okay, that's where you get your bile drainage from the common bile duct as well as the pancreatic duct and so on and so forth. Okay, so bile staining here, non-bile staining because you just have essentially nothing to pass into before things kind of come out from the liver. Okay, duodenal atresia, they love this buzzword heavy double bubble sign. Okay, keep saying it 5 times until it rhymes, but it's fantastic. It's a very big buzzword. Okay, functional pyloric stenosis is another thing to keep in mind, but don't even trip about this. If you see double bubble sign, it's going to be duodenal atresia. Okay, this one is good. Annular pancreas. So this one is a little harder to imagine, but what you need to remember is that the pancreas isn't just one solid giant mass that was created overnight. What happens is that the pancreas has actually 2 buds, right? There's the posterior bud, and then there's like the more ventral bud, right? Yeah, we're gonna go with posterior and ventral at this point. Basically, the buds that are more ventral or like more front located, what they essentially have is 2 aspects, right? Those 2 aspects are supposed to migrate backwards and join and become the pancreatic head. Unfortunately, sometimes they malform and then they malrotate and they unfortunately go around each other and then they still come together and become the pancreatic head. But they're going to cause essentially a stenosis that is more mechanical in nature, right? It's because it's just a compression caused by the pancreatic bud not being able to come together properly, and that's going to cause a stenosis. Now, the difficult thing with this is that is this going to be bile stained or not bile stained if this baby were to develop normally? That's the question that I have for you guys. Yeah, this one's a little difficult. It is going to be bile stained because bile is still going to be coming out. It's just going to be likely posterior, right? Like this is actually more, more posterior. So your common bile draining duct is going to be right here. So bile is still going to come here, but the problem is that it's still going to be bile stained. It's going to be, it can be a little difficult to conceptualize this, so just keep this in mind. This wasn't highly tested, but it is a good one to keep in mind. The subhepatic cecum and appendix. This is once again, midgut rotation that's kind of gone wrong. Remember, the rotation is supposed to happen at a particularly important kind of pathway. It's supposed to be going clockwise and then counterclockwise. So it's a little bit of a mess when you guys have to think about it that way, but I would learn it based off the clinical correlates, right? This may have a diagnosis of appendicitis because remember, everything is draining directly into one of your small intestines and the large intestines that are more closer to each other. So then you have your mixed rotation with valvulus, right? Valvulus changes may cause duodenal obstruction, right? Due to failure of the midgut to completely finish its rotation at the 90 degrees, okay? Okay, this one is very buzzword heavy. If you guys see incomplete closure of the umbilical ring and then covered by skin, umbilical hernia. It's as simple as that. You don't even have to think further than that. Omphalocele, this is important. You've got to know that it's a little different because it is a membrane. It's a fetal membrane, right? It's not, this one is surrounded by skin, right? It's covered by skin versus a fetal membrane, right? That's an omphalocele. Omphalocele is closed, right? O for omphalocele. Okay, gastroschisis, the S is for the S-shaped organ stuff that's basically protruding out of the baby. Now, this can cause hypothermia because remember, your organs have to be properly perfused with blood and kept warm for all sorts of biological and biochemical pathways to continue. This one is straight up incomplete closure of the lateral folds. So if you see this buzzword, lateral folds, incomplete closure, boom, you know it's gastroschisis. Okay, Meckel's diverticulum. Honestly, I'm tired of talking, so tell me the buzzword for Meckel's diverticulum, guys. The rules of two. I love it. Awesome. Awesome, awesome, awesome. Exactly. It can mimic, it can basically mimic a appendicitis, but it's the omphalenteric duct, right? That's what you have to keep in mind for Meckel's diverticulum. That often tends to be the question that they ask, but they will give you these in the question stems to keep it straight, right? 2 times in males, right? It's 2 inches long, 2 feet from the ileocecal valve, and then 2% of the population have it. And it's like commonly first 2 years of life. One may have 2 types of epithelium, gastric and pancreas. This part really tripped me up because I forgot that, you know, this was also a, you know, part rule of 2, but just keep this in mind because it has gastric and pancreatic tissues in terms of epithelia. Okay, what's the difference between a cyst and a duct? A cyst is basically a cavernous kind of structure as compared to a straight up fistula or a kind of communicating junction. So here it's kind of be a little sad to kind of witness because the baby's first, I guess, poop is going to be coming from their belly button, which is kind of wild because it's fecal discharge from the umbilical. So if they say any sort of meconium coming from the belly button, you already know that it's the umbilical ileal fistula, okay? And, you know, it often presents with infections because remember, your bacteria that is basically hanging out outside your, you know, skin can easily pass into the intestines and cause one, either sepsis or, you know, the baby could pass away due to the septic shock that these exogenous bacteria are bringing in. Okay. Development of the anal canal. This one's a little tricky. They do ask this question. The superior 2/3 is from the hindgut and everything that's below is the inferior 1/3 is from the proctoduum. It's basically a portion of the epithelia, right? I can't pronounce that for the life of me, but you have to remember that the pectinate line is the one that distinguishes anything above and essentially is being the hindgut and anything below being the epithelia from the ectoderm, okay? See, ectoderm of the proctoduum and endoduum, right? So just keep that in mind. Blood supply, they like to ask you guys this question in terms of the superior rectal arteries, right? That goes into the IMA versus the internal or the inferior component is from the internal pudendal artery, right? And the internal pudendal artery, guess what? It goes into the pudendal canal. Who would have thought? I love when anatomists kind of keep it straight. Innervation, above the pectinate line, autonomics, anything below, it's going to be inferior rectal nerve. And guess what? Is it going to be somatic or visceral control? Somatic, exactly, right? This is the thing that allows you to run to the bathroom when you need to, as compared to just, you know, passing stool when the urge hits you. So these were actually tested pretty heavily for us. Low anorectal abnormalities with anal stenosis, right? When you see the buzzwords for these is slight dorsal deviation of the urorectal septum, right? That is the buzzword for the anal stenosis, right? as compared to imperfect anus, right? The buzzword for this was a little bit more tricky because what essentially happened here is a blind outpouching, right? It's basically, I don't know, did they, yeah, no, that's just a blind outpouching. That's what you need to remember. Versus the membranous atresia, it's going to be failure of the perforate at the end of the 8th week. So if you don't see a perforation and there's some kind of blue kind of presentation, then you know it's a membranous atresia versus an imperforate anus. Okay. Anorectal agenesis with the fistula, that's when the pouch goes out into the, unfortunately, it's kind of scary to even imagine. It goes directly into the vaginal canal, or it goes directly into the baby's urethra, right, for males versus females. So if you guys can remember that, right, as a result of the incomplete separation of the cloaca, right, which is something you guys learned in ER. So make sure you guys know that information. Urogenital sinus by the urorectal septum is basically lost. So everything kind of goes either into the vaginal canal or the urethra, depending on male or female. Okay, rectal atresia, right? That just means you either can have one, a atresia that is just a blind, right, outpouching, right? Versus here, this is another blind outpouching where you have an anal canal, but it's just not connected with the proximal rectum, right? Which is part of the hindgut structure that hasn't developed all the way down. Okay, now to my lovely folks who have always heard about Hirschsprung disease, congenital megacolon or toxic megacolon, tell me which portion, well, is it the dilated portion that's affected, or is it the skinny portion that's affected without any neural crest cells? The skinny portion, exactly. That is critical, okay? You have to remember that. A lot of people miss those points by saying it's the dilated portion. The dilated portion is actually because the passage of stool or any sort of other thing cannot make its way. Like it's just stopped here because there's no innervation by the myenteric plexus or any sort of autonomic systems that allows for peristalsis to take place. So this is just sitting there. It's just a hollow tube without any sort of peristaltic movements. So it just kind of gets blocked up here. So everything has to kind of hypertrophy and compensate for it. So keep that in mind. They like this image as well. Okay, anatomy, peritoneum. Okay. Essentially what you need to know with this is, I'm going to keep it very simple, is you need to know this structure. Okay. You need to know your intraperitoneal organs versus your primary retroperitoneal organs. Okay. They love asking this. Does this apply for the myenteric and Meissner's plexus? Yes, I believe so. Because you don't have autonomics going in, as well as, because all of your autonomics, whether it's coming from the neural crest cells or the innervation that's coming from the central nervous system, your spinal cords, can't make its way all the way for the Hirschsprung disease. Okay. Intraperitoneal organs, you guys, I believe there's a nice little mnemonic for your retroperitoneal structures. So if you guys have this down, right, you can just basically do a process of elimination for any of the structures that is intraperitoneal, right? Intraperitoneal, stomach, liver, first part of the duodenum. This was a question on our exam. And then the small intestine, spleen, cecum, and appendix, right? Whereas your primary versus your secondary have to be kept straight, okay? It's easier to memorize with this mnemonic, right? Sad pucker, but SGU will ask you guys if it's secondary or primary, okay? So keep that in mind. Make sure you guys cover that and keep it straight for the exam. Peritoneal cavity, because remember, this is essentially going to dictate a lot of the ways pathology or any sort of disorder is going to present in terms of both ligaments and structures that could be affected, and how any sort of thing can travel from one part of the digestive system to the other. So let's kind of go over this. Okay, tell me guys, what's another name for the omental foramen? There you go. Foramen of Winslow, right? Sounds like, I don't know, like one of the dukes of, I don't know, I guess the British royalty, like the foramen of Winslow. The omental foramen is a access point to what? What is it? Lesser sac, okay? The lesser sac, let's say I have a posterior stomach, or I guess peptic ulcer. Where is all this wonderful bile acids going to, not bile acids, wonderful acid going to go? Lesser sac, exactly. What is the structure that's going to likely be damaged? What organ is likely going to be damaged below? This one's a little trickier. Pancreas, exactly, guys. It's pancreas. Okay, so remember, a stomach, a peptic ulcer, right, can actually lead to the enzymatic increase, right, found in serum of pancreatic lipases and amylases. So it's a very, very difficult question that they can ask you based off of anatomical structure, and it's a great one to really test your knowledge of where everything is situated. Okay. Let's kind of go over a couple of the things. Greater omentum has the gastrosplenic, gastrophrenic, gastrosplenic, gastrocolic kind of components, and they basically tell you what makes up each of the regions and boundaries of it. Okay, so the other thing that they like to ask, hepatoduodenal ligament, it's their favorite for the Pringle maneuver. I don't know why a box of chips has to do with anything, but apparently it helps prevent blood from, going to the liver, and you can do kind of hepatic transplant or sort of maintenance of any sort of structure that's there. Okay. Boundaries of the omental foramen that you need to keep in mind is the hepatoduodenal ligament, right? That deals with the portal triad. Inferior, right? The duodenum. And the posterior aspect is the inferior vena cava. This was actually a question. They asked us, like, what major structure could be damaged that was a vein, that was a venous structure. And we kind of had to break our heads, but we realized quickly that it was the posterior aspect of this entire location. So it's the inferior vena cava. Okay, let's look at the greater and lesser sac. The greater sac, right, is the main large component that you essentially would see when you cut open someone's guts, right? The lesser is less visible, but you can access it through, like what you guys have been saying, foramen of insula or omental foramen, and it's a great way to assess any sort of damage that's happened at the stomach level, and posteriorly you can see like where everything could potentially be damaged, the pancreas, right? Because look, this is the pancreas right here, guys. Guess what? Any sort of bleed would go out, or any sort of ulcer could go directly back here and damage and lead to elevated amylases and lipases levels. Okay. I don't know why the gutters weren't that big of a thing on our exam, but I don't know if it will be for you guys. Remember, the left paracolic gutter is a little interesting because it's narrower and partially obstructed by the phrenocolic ligament. So what that essentially means is that one, if there's any sort of bleed, it will have a harder time getting in there. But if there's any sort of infection, it can quickly travel to the subphrenic space because there's a direct link that holds, remember, all these organs as well as your internal reproductive organs together in your body cavity. Because remember, nothing's ever just freely floating around in your system. Everything's tied together by ligaments and connective tissue. Except when if you have any sort of bleeding on this side, right paracolic gutter can go directly down, and guess where it'll go? The lowest most points of our system. Okay. Now, this one is difficult, right? Guys, you got to pay attention to this, and they're going to ask you questions about these pouches. The pouches of Morrison, right, is the hepatorenal recess. So this is one of the things, if there's any sort of liver level bleed and the patient's lying supine, this is one of the locations where it can pool, right? Hepatorenal recess is the most dependent space for collection of pathological fluids for a supine patient, right? The patient's laying down on their back. This is one of the locations where you can pull fluid from to assess if the patient has some kind of sepsis, if there's a bleed, if there's air in the system. It could even be assessed here. Okay, remember what I told you guys about the paracolic gutters? The right paracolic gutters, if it's like nice, and it is nice and open, and if there's a bleed, guess where it'll go? It'll go to the rectouterine pouch, also known as the pouch of Douglas, the cul-de-sac, the worst cul-de-sac in the world, because you have to do cul-de-centesis in order to assess for any sort of fluid that is captured at this point, right? So essentially you have to go and take a needle, pull out any sort of fluid that's It can also be presented with ectopic pregnancies that has ruptured. So keep this in mind. That's an ER cumulative knowledge. For your males, the equivalent point is the rectovesicular pouch. Obviously, this is a little harder to do, and you'd have to go through the rectal canal in order to pull out fluid. Most of the times, they don't tend to do this. There are other points that they can pull fluid from in order to assess the patient. And it's only tested when the patient's standing, okay? For females, it's a little different. You could actually have them being supine and still assess for the pouch of Douglas. But if they ever ask you for females, and if they're standing, rectouterine pouch or the pouch of Douglas. Okay. There's a couple of like narrowing points at each of the esophageal area. So there are a total of 3 that you have to keep in mind where there's narrowing, where food or anything can kind of get stuck. This is basically what you have to keep in mind because you can basically take an image and see where the patient has issues. So 1, upper esophageal sphincter is a great one that they can ask you. The second point is where the esophagus crosses the aorta. And the third is where essentially, is at the esophageal hiatus. That's because you are essentially penetrating through, because you're penetrating through the, I swear I'm losing my brain. It's penetrating through the diaphragm. There we go. I couldn't pronounce words for a second. But when it penetrates through the diaphragm, right? Remember, you guys remember I ate 10 eggs at 12. This is one of the great structures, great mnemonics that helps you maintain that knowledge cumulatively and tells you at level 10, it's where you're perforating through, right? So that's going to be a narrowed out space. So food can also get stuck there. Okay. So these are the places that they'd like to test. We got a question on our exam. The muscle of the esophagus are varied, right? Like the skeletal component is the first 1/3, the mixed is the middle 1/3, and then smooth is going to be the lower 1/3. Okay. Arterial blood supply. Okay, you guys just have to remember celiac trunk. Okay, that is the main component, and everything that comes for the foregut basically branches off of the celiac trunk. So what are the things that they like to ask you guys about? They like to ask you about the celiac trunk. They like to ask you about the left gastric because it is a direct branch that comes and goes 1 up for the esophageal branch as well as the 2. So you guys ever wondered why, like, you could have like red stained blood when patients vomiting? It's something that could potentially be ruptured with the esophageal branches, right? There's also one of the ones that they love to ask in terms of a vasculature that's damaged with peptic ulcers. It's the splenic. Fun fact. So if you get a question, what vasculature is damaged based off of like a peptic ulcer that has perforated from the back, it's the splenic. Okay. The splenic is really important because it goes to the 1 spleen, but it also comes down and loose and creates kind of the descending aspect of the greater curvature. Okay. On the left-hand side. What else did they ask us about? We had a question on the common hepatic as well as the gastroduodenal. Guys, if there's a rupture, right, or a ulcer through the bulb of the pyloric region into the duodenum, what artery is going to be damaged?
Sorry, can you say that one more time?
Yeah, if the duodenal region has a ulcer, right, and your stomach acid is floating through, what vasculature is likely damaged? There you go. Posterior, superior, pancreaticoduodenal. Most of the times you don't have to go that deep, but I would just say gastroduodenal and you can make your peace with it, okay? Just keep that in mind. They do like to ask about the hepatoduodenal artery because it's the main branching point for all of the ones that are going to the more foregut structure of the duodenum. So once you have that straight, you should be okay. There you go, perforated peptic ulcer. Okay, if you guys ever see this, this is not a shadow, okay? If you see a shadow on the left-hand side of the patient, what is that? I'm just kidding. The patient has to burp if it's on the left-hand side. Versus here, if it's on the right-hand side, that means that there's some kind of peptic ulcer and any sort of air that's being captured is now going and irritating the diaphragm and the phrenic space in that area. And guess what, guys? Where does the pain appear? Where does the pain appear if it's in the phrenic area? Shoulder, exactly, okay? So you guys have to know the clinical signs of this as well as the radiograph image that they might likely give you as another supplement in your question steps, okay? Here you go. This is what I was telling you guys about. An ulceration / perforation, right, can lead to, in the posterior duodenal bulb, right, of the gastroduodenal, that's likely damaged, okay, of the gastroduodenal artery. You guys gave me the longer one. That's that's even more proximal. Don't even trip, but just keep it simple. Gastroduodenal for here. Small intestines. Okay. We're going to cover this in great depth, but all you need to know at this point is that it's duodenum, duodenal, jejunal junction. Then you have your jejunum. Then you have your ileum, and then your ileocecal junction. You need to know these anatomically because there's also absorptive components that you guys are going to cover this week with a couple of different professors. Okay, now, if they ever ask you, right, the major duodenal papillae, right, there's another name for it. It's called the hepatopancreatic duct or the duct of Warshung. It sounds like Worcestershire sauce in my head, so I don't know. I thought it was kind of a dumb way to remember it. But it tells you that at that point, you're in the middle ground between the foregut and the midgut. So anything above this is foregut, anything below, midgut, okay? Another thing to remember is that the common pancreatic duct also drains into this point. There's also an accessory component that goes in and goes to the minor duodenal papillae. No one really cares about it for now, but just keep that in mind that it could be an opportunity for a question. Remember, the aorta as well as the vena cava and the components of the superior mesenteric vessels run above the transverse duodenum. So any sort of food that's stuck there or any sort of changes that happen at the transverse duodenal area can lead to vasculature that affects downstream intestinal stuff as well as potential systemic presentation, okay? This one was asked about for one of the questions, which was the ligament of Treitz. I thought it was like the ligament of Trotz. I thought it'd be a cooler name, but it basically is what holds us up when we're going through hard times, like Trotz. Just kidding. Ligament of Treitz holds up the descending portion of the duodenum, right? Especially when you're going into the duodenal-jejunal junction. It's basically held up by this, but component that you have to keep in mind is that if there's any changes, right, with the esophagus, you can have downstream effects on the duodenum, okay, especially that junction. Okay, jejunum, if they say feathery, go with jejunum, okay? If they say Peyer's patches, go with ileum. That's as simple as that gets. They might give you these images just in case, just to keep it kind of rock solid, but feathery, jejunum, any sort of like Peyer's patches, go with ileum. Okay, jejunum, all you need to know is long basa recta, okay? For the ileum, it's short basa recta. That's essentially of the major kind of questions that they like to throw at you guys, but remember, all of these are coming from what? SMA. Very simple, nothing too crazy about it. Appendix. What do you need to know about the appendix? Remember, first off, it's going to present with, oh, my tummy hurts in the belly button region, right? T10. That is visceral pain, right? It's very like non-general. It's like everywhere. It's like everything hurts. But when you get to somatic pain, right, it's localized. That's when it becomes problematic. You can even have pain in the back because I think like your psoas muscles get damaged too by the inflammation. So one rebound test is for tenderness, psoas sign, and then McBurney's point. So if you guys remember that, you guys are solid. Small bowel obstruction, buzzword, stacked coins, all you need to know. Here, it gets a little weird. You might see a little bit of increased peristalsis, but it's harder for to distinguish that. Don't trip. If you see stacked coin appearances or this radiograph image, which they directly threw on our exam, that's what we got. Okay, house stress. It's basically the segmentation that tells you about the large intestines. And how do you know that you're in the large intestines? If you were a surgeon, you would look for the Taenia coli. So that was a question on our exam. Okay. Everything else you guys can literally read on your own because it makes so much direct sense, because it's like the ascending colon, there's the transverse colon, and then there's the descending, there's the sigmoid where everything fecal matter gets stored. It's also the site where most often you're going to get diverticulosis, and if it gets bad enough, it'll become diverticulitis. Okay, now you've actually spent some time in the ER module actually learning a little bit about this, I believe. So what you need to know is that anything above the pectinate line, anything below the pectinate line, you guys just need to remember endodermal, right? Visceral, afferent, right? If they have any sort of, let's say, hernias or ulcers in the above the pectinate line, it's going to be very visceral pain, right? It's very like just tenderness, like non-localized. It's not very sharp. Whereas everything below the pectinate line, it's somatic pain, because remember the pudendal nerve is just going like, dude, what are you doing? Like, you need to have more fiber in your diet so that you don't get all these weird problems. Okay. Obviously, you guys have covered this a little bit with autonomics, internal anal sphincter, right? It's governed primarily by SNS and PNS, right? SNS constriction, PNS relaxation. What you actually have control of, somatic control of, it's the external anal sphincter, like most things, right? Same thing what happens in the bladder too, guys. External sphincters often are controlled by somatic, so they have striated muscles versus smooth muscles that you see with internal. So what do you use it to control with? Inferior rectal nerve, branch of the pudendal nerve. It's all very like knowledge that's coming back for a full picture. Arterial supply, what you need to know, okay? This isn't too crazy. You just need to know that there is essentially two major components: the superior rectal artery branch that gives off branches of the inferior mesenteric, right? And there's the middle rectal artery, which is from the internal iliac, and there's the inferior rectal artery, which is the branch of the internal pudendal. So it's very similar to what we looked at above. It follows the same kind of thought process, so you don't have to get too confused with this. This is the part that gets a little trickier with lymphatics. Anything above the pectinate line goes to the internal iliac. Anything below, horizontal inguinal, guys. I swear, the only way I remember this is like when you're like sitting down and I don't know, like crisscross applesauce, it's horizontal, right? Like you're sitting, hopefully you're sitting down horizontally, not like vertically or something. So that's how I remembered superficial horizontal inguinal versus internal iliac for anything above the pectinate pain line. Remember, anything that happens at the vasculature in terms of drainage, right? It's either going to cause one, it's going to, I mean, obviously it's going to go back into the inferior vena cava, but something to also keep in mind is that there is components that's involved with the portal system. So if there's inferior mesenteric vein that's kind of messed up, it can have downstream effects. So portal hypertension can have presentations at the anorectal canal. Okay, and what's going to be likely affected? Superior, middle, and the inferior rectal veins become varicosed, right? Varicosed. That's the official term for blood in poop. Okay, I love all the technical terms I'm using today. Diverticulosis is basically outpouching. This actually happens commonly. It's not something like wildly out there, but it does cause painless rectal bleeding. If it becomes actually infected, then it becomes diverticulitis and itis, remember inflammation and problems. It's basically obstruction leading to infection. It can lead to rupture. It can have a lot of downstream presentations of like lower abdominal pain, abdominal tenderness, fever. It's very generalized, but what you need to remember are the terms of malinia, hematoschisia, and the meconium, right? Which are all terms that you guys have come across before. Okay, guys. What's the buzzword for sigmoid volvulus? Buzzword, coffee bean. Exactly, twisting and coffee bean. That's all you need to know. Double loop obstruction and coffee bean sign. So why does it look like coffee bean? I think Dr. Marshall might have drawn this out for you guys. It looks like a coffee bean. I guess that's how coffee beans look like. I've never seen one in my life because Starbucks exists in America. Okay, arteries of the intestines. So you guys can spend goober amounts of time learning about this, but you don't need to, because the major thing that they like to ask is about the watershed area. So if you guys look at this, right, everything here has its own direct blood supply as well as collateral blood supply. But what you need to remember that the SMA and the IMA have essentially points that give off blood to the left haustra, or like the flexure that you see of the large intestines. So what's it likely going to be giving? What are the two arteries? If they ask you very specifically, it's the middle colic, and the other one is going to be the marginal, right? So if you guys remember these two, then you guys are going to be good, okay, in terms of what is giving collateral blood supply to the haustra. Or if the SMA is blocked, what's likely going to give some blood to this portion? It's going to be the left colic, marginal. Both of those work, okay? Venous drainage, okay? Very simple. It's named after the location that they're often going. Splenic vein, okay? Inferior mesenteric vein and superior mesenteric vein. I swear, it doesn't get easier than that, because most of the times anatomists love to make things complicated. Okay. Look, lymphatics, they're not getting too crazy with this because they're named after the location that they're often dealing with, right? Celiac nodes, guess what? It goes with the celiac artery as well as the celiac vein, right? So it's not too crazy. Superior mesenteric as well as inferior mesenteric all have kind of components. But the key thing that they might often ask you about is where, if there's a cancer, where would it likely present? Cisterna chyli, it's called the pitcher's milk, right? You guys learned about it in term one, I'm going to say, and it's basically the accumulation of any sort of lower thoracic or even components of the leg, right, of your legs, any sort of cancer or anything that can metastasize will often use the lymphatics to kind of go where they need to go. Okay, surfaces of the liver. Okay, they're just essentially two things we got from this was that there is a gastric impression where the stomach basically just leads an indentation. There's a renal one, and then there's the colic impression. But what do they ask you about the porta hepatis? You need to just know that the common hepatic duct, the proper hepatic artery, and the portal vein are all components of this porta hepatis. Okay, everything else is basically superfluous information, not inflammation. And the only other buzzword that I remember was subphrenic space being tested for any sort of issues with liver or any sort of perforations that can lead to presentation of like shoulder pain on the right-hand side, okay? Artery supplies, you guys know this. You guys have probably covered this to death. Remember, the liver isn't getting the richest blood supply ever. It's mostly getting secondhand blood via the portal system, right? So everything that comes in isn't super oxygenated. And you guys are going to cover this when you guys are covering, I think, liver histology. There's like different zones and they can have different presentations, okay? Liver produces large amounts of lymph and mainly goes to the celiac nodes, because remember, liver, guess what? Foregut. So celiac nodes, very simple. Okay, biliary hepatis. Really, the only question we got on this was the common bile duct, right? If it's shut down right here, right? Everything gets backed up, and that's going to lead to, right, any sort of elevation in the liver enzymes that you guys know, which is commonly seen, is going to be elevated ALT, AST, right? Those are the things. The ratio is going to be more dependent on if it's like, if you have any sort of bile obstruction via chronic cirrhosis caused by alcohol. Obviously, pancreatitis could be also involved with this, but don't think too much on this. Everything that you see here eventually drains into the major duodenal papillae. Okay, biliary structure. I'm gonna not spend too much time on this because this is something that comes back again in T4, but all you need to know is that there are these kind of components that I'm highlighting here, which is the common bile duct. You need to know the main pancreatic duct, and then you need to know the accessory pancreatic duct, and that there are two papillae. Guess what? There's a minor one, and then there's a major one. And no one really cares about the minor, so only questions you're going to likely get are about the major one, okay? Guess what? Right upper quadrant pain. And unfortunately, I think Dr. Marshall or someone, I think, remember, they said the buzzword is fat, fertile female of 40 years of age, which I kind of sounds like profile-y to me, but I'm just going to roll with the punches as they come, okay? Okay, this was a question we were asked. They do ask you guys, and they will outline all of these for you, and they will ask you what the structure is. So it's a very first order question. If you guys can remember and go through this, you're going to be completely fine. Okay, This one, we were asked all the way up till our BSCE. So make sure you guys learn this very well, the boundaries of the triangle of Calotte, okay? I don't know why, they just love it to death. But what they do is the liver serves as the superior boundary, right? The lateral kind of component is the cystic duct, and the medial component, right here, is the common hepatic duct, right? And what do we use to find it? The content that we're looking for is the cystic artery, right? It's apparently the thing that they keep on wanting to test over and over. So just keep that in mind, cover it very well.
Hey,
Okay,
Kish.
sorry,
Yeah,
what's up?
Quick thing. Can you just like go over the watershed area? Some of the students were asking about that just really quickly.
Yeah, no problem. Let me pull it up. Okay, so this is the watershed area right here. Remember, the questions that they likely ask you is middle colic versus marginal, right? All you need to remember, if marginal isn't there, middle colic is your next best answer. That's going to give blood supply to the watershed area. So splenic flexure, okay, if there's a blockage, right? If let's say SMA is occluded, what's going to likely give blood supply? Marginal.
I'm sorry, can I ask you a quick question about that?
Yeah,
So
go
you
for
were
it.
saying if that was the collateral blood supply to the hostra, and then also with the SMA, I know that the left colic, like I know that I asked her in the chat about the left colic. So can you just brush over that part really quick?
Yeah, so it's a little challenging with this because we were often asked about like middle colic versus like the left colic. Oops, my marker went away. Left colic versus marginal. The marginal is what actually makes up the line that is created here. It's essentially the first kind of like line that gives blood supply to this this splenic flexure and the watershed area. When they ask you a question, it likely is going to be based off of middle colic, either the left colic or the marginal, because these are the major kind of suppliers of this region. There's no independent supply that goes directly to it. So let's say one of this is occluded, right? Let's say the SMA is completely gone, right? What's likely going to give you some collateral blood supply, right? Some anastomoses. It's going to be either the marginal, right? The marginal is the first most line. And typically they don't like to ask that one because it's very generalized because it makes up the entire kind of line that you see here. That's the marginal. But the next best is the left colic, and the left colic goes up like this. It's a little, because remember, it has to work against gravity, so it's not going to give you as much blood, but it's still sufficient enough to keep you going. Did that clarify it?
Yeah, thank you. I'm so sorry. I kind of like what made you go backwards, but thank you.
Yeah, no biggie. It's best to answer those questions because I'm pretty sure someone else had that question too. So I am more than happy to do it. No problem. Let's go over some of the spleen and blood supply that's going to the pancreas. Okay, so you guys are probably thinking spleen. Yes, spleen, splenic artery is very important. It's one of the branches that comes off of the celiac. Obviously, it's critical because the spleen does a lot of blood filtration and making sure you guys get rid of all sorts of like, obviously there's also kind of presentations with immuno, but we're going to cover that in term 3. What you need to know is that the artery is also giving off multiple branches to not only give blood supply to the pancreas, right? The pancreas, the tail of the pancreas actually is the location where you produce majority of your insulin. So if there's any sort of insulin level issue and you had to like take out like, let's say like the spleen, you got to make sure you preserve some of the vasculature going to that because the patient can present with, hyperglycemia because the insulin can't be released anywhere. It just sits there and doesn't know what to do with itself. So any sort of splenic rupture can also kind of present with multiple damage to organs. Blood can pool downwards. What you also need to know is that splenomegaly can also cause like so many other things. But let me show you guys a picture of this. Is actually there was another picture that actually showed this, but let me see if I can find it. I'll cover it, but essentially the splenorenal ligament, right? If you occlude it, you also have to make sure not to like mess up the tail of the pancreas, okay? It's often done if you need to remove the spleen and you just have to be careful not to mess with it because the Also, the splenorenal ligament is actually attached to the pancreas too. So got to be very careful with that. It can lead to sort of pancreatic downstream issues. Portal vein, okay? It's basically a collection of everything that you're capturing from multiple other locations. It all deals with the filtration process done at the liver. You guys are going to see this again in the next week material. Don't trip too much on it because they're going to give you the actual high yield components. But the questions that they like to ask, and we were asked multiple times, was hematemesis. It's when you literally start coughing up blood, like it's scary if you guys have ever seen this, but it's projectile vomiting of blood. It's wild because of esophageal varices. It's dealing with a little bit with portal anastomosis, right? The esophageal tributaries of the left gastric and the esophageal tributaries of the esophagus and the hemiesophagus basically meet up right here. And there, if there's any sort of tension or any sort of issue that affects the liver, then it can lead to rupture of these said vessels and lead to blood into the esophagus. And at the esophagus, they can then throw it up because it's a reflex. Okay, it's a very buzzword heavy questions. Hematemesis, no gastric vein, no esophagus and hemiesophagus. Okay, now this one wasn't asked too much, but it's dealing with hemorrhoids, right? It's the superior rectal vein and the inferior and middle rectal veins, because remember, that's coming from the internal iliac vein versus the inferior mesenteric veins that is likely being tested. Okay, hemorrhoids is a common thing that happens to a lot of older individuals. Okay, caput medusa. I swear, if there's one thing you guys take away from this, caput medusa from anatomy, because they love this question. Paraumbilical veins, tributaries of the superior and inferior epigastric, because remember, it's superficial. The medusa like kind of presents at the stomach level, and it just the patient's stomach looks ballooned out. They're dilated and tortuous, right? And they're superficial epigastric veins. Superficial epigastric vein and paraumbilical veins is what you need to take away. Retroperitoneal organ kind of issues, they don't ask about this often, but it's the veins of the colon, duodenum, pancreas, and the spleen that deals with the renal, lumbar, and esophagus vein. Not often asked about, so don't even trip about that. Okay, pancreas, this is what I was looking for. Oh my God, there it is. Right, so the vasculature you guys see is the splenic artery that's going like this, but it also descends downwards and gives rich blood supply with the gastroduodenal components that's going down this way as well as this way. So it has a rich amount of blood supply, but remember the tail, the primary blood supply is the splenic artery. Okay, most posterior to the stomach parts of the head, it's the uncated process, neck, body, tail. That's what you need to remember. Mobile within the splenorenal ligament. Okay, they like that question a lot. Any other pancreatic stuff? Yes, anterior, posterior, superior pancreatico-duodenal arteries. There it is. That's what I was looking for. Branches of the gastroduodenal artery, right? It's coming off of the celiac artery. Now, remember, it's dual blood supply. Anterior and posterior inferior pancreatico-duodenal artery is coming from the SMA, and the pancreatic branches is coming from the splenic artery. Clinical correlates, right? Rupture of the pancreas, bad, really bad. You can also have pancreatic cancer, right? And that's something you guys are going to cover in T4, so don't even trip about it. It's mostly dealing with rupture of the pancreas and what it's going to present with, which is elevated pancreatic amylase and lipases. Okay, this one, I'm not going to insult your guys' intelligence. You guys, it's mostly labeled for you guys. So you just have to go through all of the clinical correlates and the likely questions they can ask about this, but it's just going over the large intestines, small intestines, branches of the SMA, because they like to ask this question based off of clinical correlates. Guys, remember SMA, it's also dealing with watershed area too. So run through this. You guys need to know your left colic areas and the flexures and all of that jazz. This one was asked about. Don't mistaken this for anything else, because this is the stomach. Remember, you're looking at this at the patient's foot level. So this is looking foot first, not at the head first. So make sure you anatomically orient yourself. Pancreas, splenic artery, right? Important right here. And you guys can see the celiac trunk right here. Look at all the little branches it's coming off of. Not really little, but it's pretty darn decent. Then you have your portal system and the splenic, and then we get into the biochemistry of it. Let me see. Let's do, I think this, and then we'll take a quick break. We'll take a 5 to 10 minute break, and then we can hop right back. Okay. Postprandial, right? Postprandial is essentially you've had a nice, juicy, yummy, delicious meal. What is happening is glycolysis and glycogen synthesis is activated, right? Everything else, gluconeogenesis and glycogen degradation is inhibited because remember, insulin is ruling at this point. High glucagon level, right, is when you're starving, right? You're like, oh man, I'm hypoglycemic. My brain needs energy. My skeletal muscles needs energy. My RBCs need energy. So what's going to happen? Hepatic glycolysis and glycogen synthesis are inhibited. Gluconeogenesis and glycogen degradation are activated. I'm not going to insult your guys' intelligence. I've already gone over this entire pathway. Here it is again. Make sure you guys know your let me get my marker, hexokinase versus glucokinase, because remember the L in glucokinase is liver. Okay, so keep that in mind, keep it straight, because they like to ask this question a lot about hexokinase versus glucokinase. Glycolysis is inactive. Glucokinase is mostly in the nucleus, right? This is a very buzzword heavy thing. Glucokinase needs to be in the nucleus, right? Bifunctional enzymes are phosphorylated, and the PFK1 is inactive ATP inhibition due to fructose 2,6-bisphosphatase, right? Is basically absent from this equation, okay? Gluconeogenesis is going to be the exact opposite. You guys will run through that pathway again, and you guys will understand it even more now because you have a systemic presentation of what is happening. Glycogen degradation and gluconeogenesis happens at the same time because remember, there's a timeline to this entire pathway. If it happens in the beginning, right, it's going to be likely you're going to mess up all of your glycogen stores first before you start doing any sort of your amino acids or you start tapping into your tags and other stuff, okay? Liver versus muscle. Remember, your liver will take precedence first and then your muscles will start kind of taking play, okay? Same time during fasting. Remember, muscles are going to favor you having those contractions and making sure you stay alive. Outline and regulation of glycogen metabolism in the liver. Once again, cyclic AMP messaging system. You guys know this. Remember, we talked about the whole phosphorylation, dephosphorylation being the entire pathway. Okay. Glycogen degradation is done by glycogen phosphorylase, right? Makes sense because what you're ultimately trying to do is get glucose 1-phosphate, right? What we essentially have is like one more step before you just cleave off that phosphate and you spit out this glucose into usable form that's going to be used up for ATP production at the local skeletal muscle level. Glucose 6-phosphate activates glycogen synthase, inhibits glycogen phosphorylase, ATP inhibits glycogen phosphorylase. So you guys can run through this. It makes sense ultimately because think about it this way, you want to either store or you want to use up, right? AMP wants you to start using up your glycogen stores so that you can make the sufficient levels of products to keep your muscles active and functioning. Let's see. Okay, disorders. This is where it gets high yield. Deficiency of glucokinase, guys. What does glucokinase do? Glucokinase is going to be found where? In peripheral tissues or in the liver? Liver, right? L for liver, right? Glucokinase is for liver. Awesome. Right? Mature onset of diabetes of young type 2, MODY type 2, is characterized by impaired insulin secretion from the beta cells of the pancreas, right? So you have no insulin, so guess what? Your body can't use all that wonderful kind of storage capacity that you have. So due to the deficiency of glucokinase, only high blood glucose levels larger than that leads to high ATP levels in the beta cells, which is needed to close the potassium channels and insulin release. So this is critical. You gotta run through this pathway once again and understand why it's happening. Where in the human body is it stored? Largest amount, skeletal muscles, the highest concentration in the liver, okay? This was something that they asked us. And in our exam, it was the largest amount. And guess what a lot of people put? Liver. Largest amount is actually in the skeletal muscle, okay? concentration, liver. Think about it. Your liver is like a one organ. Your skeletal muscles are pretty well distributed. So like, think about it bulk-wise, it's going to be all over the place, right? A large amount. Concentration, your liver does a lot of stuff. It's a very concentrated organ. It's focused, unlike me sometimes. Okay. Purpose and regulation of glycogen synthesis are the same for liver versus muscle, but what we ultimately end up doing is glycogen synthase is a regulated enzyme, right? Your insulin, what is it going to do? It's going to lead to activation and dephosphorylates the protein phosphatase 1. So basically a portion of your glycogen synthase just says, hey, I think I should start storing all this excess glucose floating around. Versus your glucagon and epinephrine, right? These are your, like, I'm in a starved state or I'm in a energy dependent and I need to figure my life out type of situation. That's what it's going to basically lead to inactivation and phosphorylation of protein kinase A, and eventually leads to the downregulation of glycogen synthase. Okay, you guys know this, right? Liver glycogen degradation needs always hormonal activation, right? Versus muscle, it's dependent on calcium and AMP, which are local factors, right? Think about it this way, glucagon and epinephrine, where it's coming from. What are the structures that are close to the liver? Guess what? Your pancreas and your kidneys, right? Those are both close to it. So glucagon and epinephrine are nearby, so they can quickly, like, change the liver glycogen degradation process. Versus something that's more proximal to the muscles, guess what? Your muscles are generating calcium and it's using up ATP to create AMP. So they're independent of these hormones, and they're often only dependent when they're considered for epinephrine and norepinephrine, leading to your stores kind of being activated and tapped on. Okay. You guys know this. You guys have run through this pathway before, so I'm not going to spend too much time, right? Insulin dephosphorylates. Remember, on state, right? Dephosphorylated. Off state, you're starving. It's going to, what is it going to do? Glycogen synthase, right? Glycogen phosphorylase kinase, and then bifunctional enzymes, as well as pyruvate kinase, right? Glucagon is all going to be phosphorylation GS. Okay, high insulin, low glucagon. Remember, it's a ratio. It's not just something that you need to think about it in terms of, just insulin's just being pushed out. It's all a ratio. Everything in our body runs on a ratio system. Fed state, right? Glycogenesis and then glycolysis, right? That's what is likely happening. You can have in high glucagon predicated states, you're going to have glycogenolysis and gluconeogenesis. Makes sense. As well as beta oxidation can happen. This is something you guys have looked at. This is the chain, alpha 1, 4, and then alpha 1, 6 is the branch point component. Starch, you guys have seen this before. Now, this is high yield. We were asked about this multiple times in every single block of our exam. So focus in on this and make sure you learn it well. Von Gerk's, right? What's the enzyme that's deficient? Glucose 6 phosphatase. Phosphatase, right? What does glucose 6 phosphatase deal with, guys? Walk me through it. What's phosphatase? Very important. Gluconeogenesis. Okay. What about lysosomal alpha 1 glucosidase? Okay. Pompei's disease. How I remember this one was they can also mention this as acid maltase. Okay, so Pompe disease, think acid maltase or lysosomal alpha 1 glucosidase. Those are like kind of buzzwords for it. It can present with cardiomegaly, kind of respiratory issues as well. There's a lot of different components with this, but I want to make sure you guys know the buzzwords: cardiomegaly, respiratory distress. You can also present with like limb girdle, muscle weakness, a lot of like floppy babies. So there's just buzzwords for those. Cory's disease, okay? Cory's disease is a little bit more challenging because it's a debranching enzyme, right? It's alpha 1,4 glucosidase and 4,4 transferase, okay? Know that very well, versus your branching one is Anderson, okay? If you guys need help with these, make sure you guys review first aid as well as review Dr. Trotz's slides because they're very, very high yield. And there is a little difference between them, okay? Glycogen stores. Yes, we did see question stem saying normal versus abnormal glycogen structures during the exam. We did make sure you guys cover that as well, because sometimes when you're talking about your branching enzymes, they can present with normal, and that could clue you in on a different disorder versus something else. Okay. They all do seem very similar, but first aid outlines the differentiating factors. I personally didn't like Dr. Trotz's slides for these as much, because it just felt like an enzyme and the deficiency, but it's very good to cover them in detail. And I outlined it here for you guys right here. So glucose 6-phosphatase, what's deficient for von Gerk's, right? Know which one is going to present with cardiomyopathy versus hepatosplenomegaly, right? Anderson disease, right? It's branching enzymes. So it's very buzzword heavy. Fill in the information. You guys can power through this. Von Gerk's disease, right? Normal structure, high glycogen amount, and its normal structure. This is what one of you guys asked about. What's deficient? Glucose 6-phosphatase in the liver and the kidneys. Because remember, we talked about where we get a lot of our gluconeogenesis and how we start taking glycogen and start ripping it apart. Liver and kidney, as well as your muscle. But muscle, you'll see the specific isozyme that's affected. So hypoglycemia, lactic acidosis, hyperuricemia, hyperlipidemia, failure to thrive. And what you have to do is basically keep feeding these kids uncooked, cornstarch, because that's the only thing that they can process. And any sort of fasting metabolism needs to be avoided. They got to constantly eat, otherwise it's problematic. It's like the worst case of being hangry. Pompey's disease, okay? Pompey's disease is it's acid maltase, right? Or lysosomal alpha-1 for glucosidase, right? That's the enzyme that's deficient. This is happening where? Most of the times it's happening in the heart, muscle, and liver. It's a lysosomal glycogen accumulation. So remember that if they ask you about some kind of lysosomal disease, this is Pompe, type 2 GSD. So this disease is grouped by glycogen stores, lysosomal, and a muscular disease, okay? Pompe, pump, right? Buzzword heavy, cardiomegaly and hepatomegaly, floppy babies. What are you going to find? CKMM, right? CKMM is your creatinine kinase muscle is how I remember it. And that's how you can keep it straight. Infantile, often death, later onset, you're going to have respiratory issues, but most of the times you can treat with immuno as well as enzyme therapy. Cory's disease, this one's a little interesting because it's a deficiency of the debranching. So what are you going to have? Abnormal glycogen, because you have to do the debranching process so that you don't have like weird, overly long chains, right? And the buzzword for this is Cory's disease, deficiency of debranching, abnormal glycogen, and then limit dextrose. Okay. Anderson's disease, okay? It's often seen as a familial cirrhosis, okay? So what you need to know, deficiency of glycogen branching enzymes, 4,6 transferase in the liver and the muscle. So it's gonna have both hepatomegaly as well as any sort of muscular kind of level damage as well. Characterized by abnormal glycogen structure, so too long of a chain, because remember, long glucose chains are less able to kind of deal with storage form. So enlarged liver, muscle weakness, hypotonia, cardiomyopathies, right? Anderson I think there's a buzzword for it. Feel free to drop it in the chat so that you guys are helping out your classmates. McArdle's, right? This is talking about specifically the muscle type of this phosphorylase, right? It's the muscle phosphorylase being deficient. So high levels of glycogen, normal structure, right? Hepatic isozyme is completely fine. So are you going to have hepatomegaly with this? No, right? It's going to be more muscular level thing. So what we remember is McArdle, muscle. Hepatic isozyme is normal. So the children are often tired, unmotivated, often like me after an exam. So patients can show rhabdomyolysis, rhabdo, right? Myolysis. This is talking about your muscles being torn apart after forced exercise due to lack of ATP. Serum CKMM, which is a cytoplasmic enzyme, is going to be elevated. So creatinine kinase, muscle. Hers disease, guess what? If there was a McArdle's, then there's a liver equivalent, which is just the hepatic equivalent. So hers is hepatic. So deficiency of the hepatic phosphorylase leading to high levels of glycogen with normal stores. Guess what? Your muscles are completely normal. So all you're going to see is hepatomegaly, growth retardation, and mild fasting hypoglycemia. So TARWI. Okay, this one is a little weird. TARWI is reduced activity of phosphofructokinase 1, so PFK1, and it's the muscle M subunit. So it's more specifically to the muscle and the RBCs. So the liver is fine. Once again, it's a little combination presentation here that you would keep in mind. So hemolysis often occurs. So 50% of PFK1, it happens with RBCs. Once again, here's the run through and each of the disorders of where it happens with the glycogen, all the way down to the glucose, like when you need to actually release it. So MODY type 2, von Gerk, it's going to be at the glucose level. Like you need to take glucose 6-phosphate and push it out, right? Like it's not functionally there. Now, what happens with the glycogen phosphorylase and McArdle's and HERS disease? It's all different isozymes, right? McArdle's, muscle, HERS, liver. Now, Corey and Anderson. Corey is dealing with the straight chain, right? Anderson is a straight shooter. That's, I think, how we remembered it. And then Corey deals with debranching, okay? And then, of course, when you're dealing with glycogen, right, to glucose, we have a little way of how we kind of regulate it, which is your lysozymes are involved with kind of cleaving a certain portions of those glycogen kind of chains. And when you have a lysosomal enzyme deficiency, such as acid maltase, or I think, what is it? Lysosomal acid alpha 1,4, the long name for the acid maltase, that deals with Pompe disease, okay? Now, fructose metabolism, we have dietary sources of fructose, which we see with sucrose, right? And we deal with sucrase, fruits and honey, high fructose corn syrup. And guess what? GLUT5. Remember you guys learned that? Fructose deals with GLUT5. I think like 5 is like, starts with the word F. Starts with the letter F. I'm like confabulating at this point. Absorbed by GLUT5, so 5. It helps me keeps it straight. Liver, fructokinase, and aldolase B are involved with this, and it presents with a lot of different things. And here we're going to take a little break. I will give you guys 5 minutes, and then we will continue. Okay.
You're doing
Welcome
great,
back.
Kishore.
Thanks, Priya. Like my voice is gone. I'm gonna sound like Bane at the end of this entire thing. Batman, full swing. Fructose and metabolism. Okay, so this one is one of the minor pathways. I'm gonna say you guys don't have to worry too crazy about this. All you do need to know is that dietary source of fructose just comes from the basic things that you can keep in mind, right? The usual kind of culprits, fruits, honey, right? But what they like to ask you about is specifically when you have deficiencies, okay? And I'm gonna hopefully teach it to you guys based off of disorders and not based off of like the entire pathway. It's one of the minor pathways. So the key things to keep in mind is fructose, just like glucose, you have to phosphorylate it to bring it inside of a cell, okay? Remember, there's fructokinase, right? Fructokinase adds a phosphate group. Once that's done, you have 2 ways you can kind of go to this entire shebang. You can either do aldolase B. Remember, there's aldolase A and aldolase B, one of the pathways from your wonderful glycolysis pathway. Aldolase B has a higher affinity for fructose 1,6-bisphosphate, glycolysis, right, than for fructose 1-phosphate, but it still plays a role in this pathway. So keep that in mind, because once it takes fructose 1-phosphate, it can turn it into glyceraldehyde and then go to glyceraldehyde 3-phosphate and eventually into the intermediaries of glycolysis. Guess what? Dihydroxyacetone is a ketone. So any sort of acetone in there is a ketone. So that's all you need to kind of worry about. Now, very simple, benign fructose urea, okay? So what is that? Let's break apart that word, benign. Guess what? It's not malignant. Don't worry, no neoplasia here. All you need to remember is that benign fructose urea is going to present with fructose, right? Just not being able to enter the cell. So fructokinase is just going to be floating around, right? So, fructokinase is going to be deficient. Fructose is just going to be floating around and eventually gets passed into the urine and then essentially excreted. So the liver fructokinase is just deficient. It's not metabolized. It's asymptomatic. Buzzword heavy, asymptomatic, okay? And then urine analysis is just going to show reducing sugars that is not glucose or galactose. They will tell you this, okay? You guys have to remember and pay attention to this very much when you're going through this. Any sort of detected by abnormal urine analysis is just a common thing that you'll see. This is a little bit more problematic because hereditary fructose intolerance, what are you deficient in? It's going to be basically you're deficient in what enzyme, guys? Put in the chat. Aldolase, exactly. So aldolase is the one that's deficient, aldolase B, because aldolase B is the one that takes your wonderful fructose 1-phosphate and converts it into glyceraldehyde. And if you just have excess accumulation of fructose 1-phosphate, right, you don't know what to do with the fructose 1-phosphate. So it's going to be pushed into one of the minor pathways. It's going to get converted into an acid alcohol, or sorry, a sugar alcohol, which then can have downstream effects on your eyes. So hereditary fructose intolerance can lead to hepatocellular failure. You can have jaundice, and you can have multiple other things with this. So benign versus hereditary fructose intolerance, keep those straight. It can present with hypoglycemia, follows sucrose and following like sucrose and fructose into your diet. So this is the entire pathway. When you have aldolase B, right, this is just excess and accumulation. When you have benign, it's just fructose, and fructose can be passed into urine and then just released, okay? Okay, now, why do we learn this? Significance of any sort of polyol pathway when you like manage anything with your sugars, it gets converted into other minor pathways and can lead to the accumulation of cataracts. Remember, one of the things that is very active is sorbitol. Sorbitol, when you have excess levels of it, it can have, you can have peripheral neuropathies. You can have, neuropathies, meaning basically your neurons at the periphery are damaged. And one of the periphery, if you really think about it, is your eyes, right? Your eyes have something to keep in mind is that these eyes are very osmotically unstable. So if you have like any excess sugars, you can have cataracts and other things kind of develop due to the osmotic differences. So sorbitol is osmotically active. It's going to bring in more water. It's going to lead to cataracts. It's going to damage the eyes and the retinas. So just keep that in mind. Diabetes mellitus, not good. This is why type 2 diabetes is scary. Significance of the polyol, it can lead to galactosemia, right? Elevated blood galactose, entry of the galactose into the lens. Same thing that you saw with your sorbitol pathway, right? From glucose to sorbitol, from sorbitol to fructose, right? Fructose isn't too bad. You can deal with fructose, but what you can't deal with any of your sugar alcohols, such as galactitol, which is a sugar alcohol by aldolase reductase, right? And galactitol is osmotically active. Once again, like sorbitol, it's going to lead to cataracts. So just make sure you read the question stem carefully for this, because they can trick you with your galactitol and your sorbitol pathways. Okay, galactose, right? Here's the entire pathway so you guys can kind of take a quick peek at it. Galactokinase can be deficient, so non-classic galactosemia. So what really happens is you just have excess levels of galactose and it can eventually just get excreted. But the problem is that classic galactosemia is the ultimate cause of any sort of problem, because what happens is you're deficient in GALT, and you don't know what to do with all this excess galactose 1-phosphate, and all of that gets converted into sugar alcohols, and those are osmotically active, and that's bad. Okay. Non-classical galactosemia, right? So what happens? You're deficient in galactokinase. So galactokinase deficiency, right? Lens galactitol takes those galactose and gets converted into a sugar alcohol, which then leads to cataracts, right? It's not too bad, but you can deal with this because you just have to restrict the baby's diet of milk and other stuff, right? You'd have to make sure you don't give them like dietary, like milk. And you can't even give them lactose or lactate or anything like that, and it wouldn't really do anything. Whereas classic galactosemia, right? is going to present with something a little bit more systemic. It's autosomal recessive, right? Keep that in mind. They like to ask those like T1 questions. Galactose 1 phosphate accumulates as well as galactose, and that leads to presentation of a lot of like presentation with hypoglycemia. And typically the baby will come in after the mom started breastfeeding right away, and it's a quick trigger because remember, the mom's like started feeding right off the bat, and the baby's going to have classic presentations of milk intolerance. Classic galactosemia, right? Lens, what happens to those excess galactose floating around? It gets converted into galactitol. I can't pronounce that word. So it's aldose reductase involved in that. And remember, this is osmotically active. So what do you get? Lens, water retention, cataracts, right? And then eventually it will accumulate in the brain and it can lead to neurological damage. The babies will have developmental challenges. And what you can do is measure their blood glucose levels as well as reducing sugars, because remember, galactose is a reducing sugar as compared to glucose. Glucose is not a reducing sugar. Okay, buzzword for this: liver damage, cataracts, and developmental delay. Make sure you guys pay close attention. Don't switch it up with sorbitol, okay? And your other kind of deficiencies that you would see with similar cataracts. Galactokinase deficiency, guess what? It's galactokinase that's deficient. Autosomal recessive, same thing with GALT deficiency, autosomal recessive. Just make sure that you pay close attention, and there's a nice way to remember it right here. Fructose is to aldose B as galactose is to uridyl transferase, so Fab gut. Okay. Reducing sugars. Keep this in mind. Cataracts, right? Liver disease, developmental delay, classic galactosemia. If it's cataracts by itself, non-classic galactosemia. If it's hypoglycemia and liver disease, this can be a little bit trickier. You have to pay close attention to the lab values. Classic galactosemia versus hereditary fructose. And then you have hereditary fructose intolerance that you will see with any sort of kid given like some kind of candy or fruits that's introduced into the diet later on when they're going through the stages, versus lactose ingestion. It's going to be right away presentation, 2 to 3 weeks right off the bat. Boom, the baby can't see, it has cataracts, which no baby should. And then you can also have benign fructose, which wouldn't present with anything crazy. It's not too late. If galactosemia is present within the 2-3 weeks, it is not too late. It can still be course corrected. It's not completely like detrimental. Like they could reverse it easily. They just have to make sure they take appropriate medication. You don't have to worry about that level of detail. Just know that for galactosemia, you just need to be careful for your cataracts, because those cataracts tend to be a very buzzword heavy and a lot of people get tripped up on it. Lactose is a milk sugar, and remember, it's dealt with at the brush border level. When we're talking about brush border, what part of the intestines are we in, guys? What brush border? What is the brush border? Put it in the chat. Yeah, I mean, you can essentially vouch for any of your large intestines versus small intestines. Most of it, I believe, is in small intestines, but don't quote me on that. But yes, mostly what you do need to know is that it's done at the brush border level, and some folks are deficient in the the enzymes that are deficient. It's either going to be the sucrose isomaltose and then maltose glucose. Glucose amylase are both high levels of active than lactase. So that's why most often we don't see issues with sucrase and maltase. We do see issues with lactase, because that is a very specific thing. And a lot of people often from Asian descent, as well as folks in southern aspects of, I think, Asia, are also likely have this lactose intolerance. How does lactase synthase regulated? Lactase is non-persistent phenotype. So it's the buzzword for this is LCT gene, guys. If you remember LCT, you're going to be good for that one. You can also be controlled by the regulatory enzyme MCM6, which is the mini chromosome maintenance 6. So these are the two ones to keep in mind for any sort of your lactose level concerns. What you do need to know is that lactase non-persistence, right? This is often developed, right? It's the primary adult hypolactasia, which is the primary lactose intolerance. Okay. Disaccharide deficiencies, which is the secondary lactose intolerance, it's due to the fact that the intestinal mucosa is basically damaged, right? It's caused by some kind of like celiac disease or some kind of other disorder that's messed up the mucosal lining of your cells, or of your GIT. The cells are messed up, but your GIT. Congenital lactase deficiency is the most severe form, and that's because you just don't have those LCTs and MCM6 kind of helping you create and regulate and maintain your lactase presentation in the intestines. Okay, lactase deficiency, it's not the worst thing. You can essentially avoid this because remember, the kids are just going to be fine. What you do need to know is that lactase, right, and lactose can be osmotically active. So this can present with the classic presentation. If someone here has had like any sort of like milk intolerance, they can vouch for this because like it is osmotically active. So it's going to pull more water into the luminal side, and the water can't be pulled back in because, you know, sugars have such big sway on our entire kind of system. So it's going to present with diarrhea, flatulence, bloating, cramps, so on and so forth. Remember, if this persists for a long time, the patient can present with metabolic acidosis. Okay. Cool. Secondary lactose intolerance, that's what I kind of said. Remember, it can be due to some kind of bacteria induced. It can also be due to some kind of celiac disease, right? Remember, this is a very buzzword response to gluten, right? There's a specific thing for celiac disease that they keep on liking to ask, which is gluten being the problem. So keep that in mind. Once again, here's a kind of a nice summary of it. Let's do TAG's metabolism. Okay, TAG metabolism is a little bit more challenging because when does TAG metabolism often happen? Is it going to be right away after you guys start starving for a day, or how long does it take? Yeah, it's later on. It's actually a later on presentation. It's going to be like, okay, you guys are saying a little bit over. Yeah, it's usually like it starts kicking in like 2 to 3 days after, and it's going to persist through and through if the patient is starving for long periods of time. Okay, first thing we're going to like to do, because everything's so readily available, is the usage of glycogen and breaking down of glycogen into glucose so that the body can use it. Once that runs out after the first 24 hours, it's going to be all of your other kind of pathways being upregulated. So what's going to primarily initiate this process, right? Glucagon and catecholamines. After long periods of time, your catecholamines start clicking in to this kind of whole process of starvation, and it's going to say, hey, why don't we just, you know, phosphorylate a bunch of proteins and enzymes and start this whole process. As always, we store tags on the regular. That's actually one of the things that makes up our adipose tissues as well, right? Excess fatty acids are stored as tags, right? This is how we maintain our long-term supply. Biosynthesis of fatty acids is going to be done during well-fed states. So it's what's going to be dominating here? Insulin, right? So everything gets stored in our fat cells or adipocytes. Some of it stored in the mammary glands for eventual production of milk, which you guys covered a little bit in ER, which you're going to cover again in term 4 and 5. Precursor hormones, guess what? Acetyl-CoA, CoA, which is the pantothenic acid that you guys covered in a lot of different cofactors for different enzymes, such as tender loving care for Nancy, ATP, NADPH, and carbon dioxide are all factors that you need. Site and synthesis in the cell is cytosol. Guess what? It's the cytosol. So once again, I would highlight this because they like asking questions about it all the time. So the source of NADPH from fatty acid synthesis. Guess what? We use NADPH primarily from, you guessed it, pentose phosphate pathway. It's one of the major things that's implicated when you're dealing with your conversion of your tags into usable energy forms. The other thing that you have to think about is the malic enzyme, which is NADP positive dependent malate dehydrogenase. So once again, keep that in mind. Production of cytosolic acetyl-CoA, guess what? It happens at the mitochondrial level, So acetyl-CoA is formed in the mitochondria and cannot cross the mitochondrial membrane. So it has to be converted into citrate, and citrate can then be shuttled in. And citrate lyase, once it's in the cytosol, can be converted out into acetyl-CoA and oxaloacetate. So you're probably wondering what happens after that. Actually, you don't need to learn the entire pathway. You can, but I highly suggest you don't, because there's only critical things you need to know. Overall, you need to take like palmitate, which is essentially like palm oil, right? Like the stuff that you get from coconut. What happens is NADPH and ATP can be broken down into the following steps: formation of malvalanyl CoA via acyl CoA carboxylase. Guys, if it's a carboxylase, what do you need? Put it in the chat. I need to know. I need to know this. What are the cofactors? A, B, C. I'm telling you guys, if you guys remember these pathways and the cofactors that go with it and all the kind of the coenzymes and factors, you're going to be so solid for this exam. Synthesis of palmitate by fatty acid synthesis or synthase, it basically creates what? Basically malvalanyl CoA. And for malvalanyl CoA, there's a whole big cycle. It basically elongation of palmitate by elongase and then desaturation by desaturase. And there's all sorts of components. You don't need to know it. All you need to know is malvalanyl CoA and acyl CoA carboxylase and the cofactors that you need for it. Okay. Carboxylation of acetyl CoA to form malvalanyl CoA. What you need to know is that it is a committed step. It is a one-way step. So this is rate limited. So guess what? You need biotin, you need carbon dioxide, and guess what? You need to put some energy into it, ATP. So that's how you get, but remember, when you're putting ATP here, it's an investment. Just like what you did with glycolysis, that investment is going to pay off with the huge payout of, I think like close to like 34 or 35 or something like that, different ATP and NADPH that you can eventually shuttle back into the system. Acetyl-CoA carboxylase is active when dephosphorylated. Guess what's dephosphorylating it? Insulin, right? Insulin is a primary dephosphorylator, and it's inactivated by phosphorylation because remember, you want to primarily store when insulin is dominating, and glucagon is going to phosphorylate, especially glucagon and your epinephrine, norepinephrine, all those factors. So keep that in mind. It's going to be critical. It leads to the active dephosphorylation of acetyl-CoA, and insulin will induce acetyl-CoA carboxylase. Cool. Fatty acid synthesis, guess what? You don't need to learn this entire pathway because all you need to know is that insulin is going to upregulate it. And what essentially happens is you take all these chains of fatty acids, right? And you can either get broken down into 5, 6, or 9 in human beings. And then even if you have an odd chain one, you can always convert it and change it into a different form and eventually feed it back into this tag synthesis pathway. Okay. So the reason why we have fatty acids, right, and tags and all sorts of things is because they have functional properties in human beings, right? A great example, what we covered in term 3 and term 4 is arachidonic acid. Arachidonic acid, it makes up prostaglandins, thromboxans, and leukotrienes. You guys are going to learn about those in T3 and T4, which involve a lot with your inflammation, deal with how you have a fever, and a lot of multiple other things like bronchodilation, bronchoconstriction, multiple things. So it's very important. There's also brain property protective kind of components with alpha-linolenic acid that deals with EPA, DHA, and DHA is very important in that role. So you need those exogenous sources. So none of us should be going on a full keto diet or like purely this because it would require a lot of different reworking of your metabolic systems, okay? Acetyl-CoA carboxylase, we talked about this. It's feedback inhibited by fatty acyl-CoA. If there's too much of it, it's going to tell the systems down below, stop it, you don't need any more. So the accumulation of long-chain fatty acid that synthesize tags is slow and the pool of free CoA gets low, and that can kind of deplete your pathway from going any further. Okay, beta oxidation. Guess what? We're starving right now. So this is 3 to 5, right? So 3 to 5 days, things are getting out of whack. So where does this happen? Intracellular location, mitochondria. So you take the activated fatty acid, right, from the cytosol, transport the fatty acid into the mitochondria, and you do beta oxidation with it. So what essentially happens? Activation of fatty acid, right? You need to take the fatty acid plus CoA and you need to make it into fatty acyl CoA, right? From there, you're going to use like 2 ATPs to essentially do this. And it's a very demanding process. But once you do this fatty acyl CoA synthase, it's going to make it into the inner mitochondrial membrane. And that space is going to then allow for you to shuttle it and make it more, favorable to be brought into the mitochondrial matrix. So what happens is you do carnitine shuttling, right? CPT1, it's going to be carnitine at the outer mitochondrial membrane. I'm going to tell you guys this. We were asked questions about where these shuttles are located versus the products where they're going to be generated and put in. So make sure you guys pay attention to CPT1 and CPT2 because they're in different membranes, but their products are going to different locations, which are intermembranous space versus the mitochondrial matrix. Okay. I'm going to run through this very quickly because you don't need to know too many things other than the fact that when you're going through this, the product that is generated ultimately that you're doing with the carnitine shuttle is acetyl-CoA. And guess what? Acetyl-CoA, once it's generated from the fatty acyl-CoA, can then be converted into either one, ketones, right? Ketone body synthesis, or it can go directly into the TCA because guess what? Acetyl-CoA that you generated from fatty acids is the exact same acetyl-CoA that you generated from your pyruvate to pyruvate dehydrogenase to citrate and all of those TCA products. So make it a whole picture, guys. Malonyl-CoA is formed in the cytosol during fatty acid synthesis and inhibited in CPT1. So this ensures that the fatty acid that is synthesized is going to stay in the mitochondria and doesn't allow for things to kind of like be moved out from where it's supposed to be. So beta oxidation is what you're doing. And guess what? The amount of ATP that you did minus the investment of 2 ATPs, look at how much you're spitting out. 129 ATPs per single palmitic acid, which is freaking bonkers, right? So that's why when you do long-term kind of, I'm not recommending everyone starve, but like I'm saying like long-term fatty acid metabolization and breakdown is going to lead to a lot of depletion of your fat stores. And it's one is a way that a lot of people try to lose weight. It's a poor science because it is often recuperated back after you start going on a regular diet. Okay. Now, SGU really doesn't care about your LCADs or your SCADs. They really care about your MCADs. Okay. So these are your medium chain kind of acyl kind of groups that is going to be mostly kind of covered as your disorders. MCAD deficiency, autosomal recessive, right? It's going to present with what? Hypoglycemia and hypoketosis. Because remember, hypoglycemia, you're not going to be able to do gluconeogenesis, right? Because you're in prolonged fasting state. Hypoketosis is because you're not creating acetyl-CoA in order to create ketone bodies in even more prolonged long state. So age of presentation, very early on, 6 to 12 months, severe, right? Metabolic crises, medium chain is what is implicated here. So when you're going through this, guys, test yourself, do practice questions because it's a struggle. Now, carnitine deficiency is very simple. It's either carnitine deficiency 1 or 2. And most of the times it's going to either have a systemic presentation versus myopathic, right? Muscle specific. Okay, let's do CPT1 deficiency. CPT1 deficiency is going to lead to hypoglycemia and hypoketosis. Guess what? Because nothing is able to get in properly to the mitochondrial matrix where they can be broken down via fatty acid metabolization, right? So CPT1 deficiency is going to be just systemic form. CPT2, right, is cardiomyopathy and muscle weakness. It's often the myopathic form, okay? Lipid deposition, triglycerides in the skeletal muscle, because they don't know what to do with all this tags and fatty acids floating around in your system, okay? Management of fatty acid oxidation, what you got to do is basically manage the hypoglycemic state by always giving them glucose. They got to constantly eat, and they have to avoid starving for prolonged periods of time. And guess what? The same things that bodybuilders take with carnitine, you got to give those supplements to folks so that they can at least have some basal level activation of the beta oxidation, as well as the fatty acid synthesis and breakdown. Okay. You can also induce this. Guess what? You can also induce a similar kind of presentation, because what happens is Jamaican vomiting sickness, there's a key fruit from the Caribbeans and Africa that basically causes the accumulation of hypoglycin A, and that inhibits MCAD, and MCAD being inhibited can no longer be able to break down that fatty acyl palmitic CoA into acetyl CoA, leading to all the same presentation that you would see: hypoglycemia, coma, and death, right? Medium chain acyl carnitines are found in the urine. Okay, oxidation of odd chain. This is a minor pathway, so just keep this in mind. Guess what? Propionyl CoA, right, is an odd chain fatty acid, right? It's only 3 carbons long, because remember, we start cleaving anything that's not even chain fatty acids. So once we start cleaving it, once we start cleaving it, what essentially happens is we break it down into components that's workable, and eventually create products that we can feed directly into the TCA cycle. So propionyl CoA, guess what? Propionyl CoA to carboxylase, what factors will it need if it's a carboxylase, guys? Exactly, ABC. Look at that. It's the same trend again and again. Now, what happens after that? Methylmalonyl CoA is going to be present. Methylmalonyl CoA needs to be converted into succinyl CoA. And guess what? You guys know your TCA cycle. So where can this enter? From succinyl CoA. So what step is succinyl CoA that's going to allow? So what from succinyl CoA, what do we go into? What's the product from succinyl CoA into the TCA? Succinate. See, you guys are tying those concepts together. Make sure you guys know that. What's the enzyme that's involved with? It's succinate thiokinase. Okay, so make sure you guys review that. Tie it together. It's a little bit difficult, but once you guys get it down, it's going to be solid. Low insulin glucagon ratio is going to be important in this. It's hormone sensitive lipase. So make sure you guys remember that it's a hormone sensitive. When you have your catecholamines and glucagon and everything kind of build up to a sufficient enough high ratio, your body takes over and it just can't do anything else. So it just starts activating beta oxidation, carnitine, and all that pathways dealing with shuttling it into the mitochondria is all upregulated, okay? Here is the full rundown, right? That covers all of it. And then ketones are a favorite. Now, before we get too far into the ketones, let's go over Zellweger syndrome. Zellweger syndrome, I can't pronounce this properly. It's basically the accumulation, and it's going to have a lot of neurological deficits, right? They're going to have hepatomegaly, hepatocellular failure, as well as like seizures and all sorts of other stuff. So make this a kind of a buzzword heavy thing. It's a defect in the peroxisomal disorder, right? They're going to have seizures. They're going to have early chances of death. And it has a lot of like neurodegenerative presentations as well. Now, alpha oxidation of branched chain fatty acid is Ruffsum's disease. And this was something that a lot of people ignored, but the buzzword for this is phytanic acid, right? In dairy products. And that's the problem that we need to keep in mind, okay? Here is the whole kind of shebang, fatty acids to beta oxidation, right? We can either go from beta oxidation to acetyl CoA. From acetyl CoA, we can either feed it directly into the Krebs cycle, TCA cycle, or whatever you call it, or we can go directly into ketones, right? And that's how we kind of run through this other system. Now, this one, I found it a little better to just draw the entire pathway. So what you do is you take 2 acetyl CoAs, right? You smush them together into acetyl CoA. And then what you need to know ultimately is the mitochondrial HMG CoA synthase, right? That's the first essential thing. And the way I remember it is HOMG, I'm starving. So that's telling you like HOMG, which is mitochondrial HMG CoA synthase. S is for starving, the synthase, right? Kind of component. From there, you have HMG-CoA synthase essentially, and from HMG-CoA synthase, you create acetoacetate, right? All of this is happening at the liver, okay? The goal is to create acetoacetate, which is the first ketone in your entire pathway, right? This is what I'm kind of outlining here, guys. Sorry, I forgot to kind of scroll down. What happens is a way that we test for this, right? Acetoacetate. The way I remember that you know a patient is in ketoacidosis is I pee to acetate, right? When they pee out acetoacetate in the urine, you will see that it's the first, ketone body that's created in the liver. When this is done, what happens is essentially it can either be created into acetone, and guess what? It's the same darn acetone that folks, whoever wear nail polish and need to remove the said nail polish, they can actually use acetone to remove it. And I think it's also used to kind of clean out beakers in the lab setting. What is actually more important, right? Once this gets blown out from the liver, or sorry, from the respiratory system, what you need to remember is the 3-hydroxybutyrate, okay? 3-hydroxybutyrate is the critical ketone that is used by the rest of your body to create sufficient levels of acetyl-CoA that can be fed into the Krebs cycle. What you need to remember is that this is all done at the liver, okay? If this is not, if they ask you those questions, you've got to be rock solid. Utilization of ketones, where does it get used up? It's used up in your skeletal muscle, cardiac muscle, and the brain, because they all need constant supply of glucose, right? And the glucose, if they don't get it, they can use ketones to get converted into, and heart muscles. Sorry, I forgot to put heart muscles. Yes, heart muscles as well. What you need to remember is that they have a specific enzyme that's at the periphery that can be used to convert acetoacetate into acetyl-CoA and then two acetyl groups. And that's called thiophorase, okay? Thiophorase is the critical one. The full name is succinyl-CoA acetoacetate coenzyme transferase. Remember, this enzyme is only found in the tissues and not found in the liver. If you guys can remember that, it's going to help you a lot. How I remember it is real basic cells can't use ketones, right? So red blood cells, a brain, and then cardiac muscles, right, are the real basic cells. So that's how I remember it. The R, B, and C. Okay. Mechanism of ketone starvation. Here it is. This is all kind of like a run through and a broad overview. But remember, acetyl-CoA can directly enter the PCA cycle and get converted into citrate via oxaloacetate combining. So just keep that in mind. Here it is. Look at the starvation time, right? Look at when beta-hydroxybutyrate gets increased as well as the acetoacetate. You guys can see the market point is like starting here and they go up. Fun fact, we were given this exact same diagram on our exam, and then they asked us what's going to be elevated based off of like one of these, and they were like, tell me which enzyme is going to be upregulated, and we had to pick mitochondrial HMG-CoA synthase. Guess what? HOMG, I'm starving. Okay, keto diabetic acidosis. Guess what? All the buzzwords from term one are going to come back in full swing of things. Kussmaul breathing, right? They're going to have fruity odor due to the fact that it's acetone being blown out, and it's called anion gap metabolic acidosis. So it's going to have increased levels of, all sorts of ketones in both the serum as well as urine. So guess what you're going to find in the urine? Acetoacetate, IP2 acetate, and that's the way I remembered it. Okay, I'm going to take a break and give you guys another second to ask questions. Let me pause the recording. Let me hit resume. Okay, welcome back, guys. I hope you guys had a quick, short break. Let's do histology. This one was surprisingly, I wouldn't say it's low yield, but it was tested as a kind of a compound of what's given in the question stem. So they're not going to give you just specifically one histo and just tell you to like tell me the function of it. So just keep that in mind. I also do apologize in advance. I put a little bit of notes, my notes that were on top of on the slides. So just keep that in mind. If you guys want to write over it, you guys can add your own kind of bullet points. You guys can. Lingual papillae, just keep in mind, Fun fact, cats actually have a lot of lingual papillae, and even crazier fun fact, tigers have very sharp lingual papillae that are basically like mini razors on their tongue. It actually helps them grab onto food and stay within their mouth. And it's also the same kind of function that it has in our mouth. So just keep that in mind. It's highly keratinized. It's not anything involved with actual taste sensation. Its primary function is just a mechanical one to capture or even pierce the food. I mean, cats obviously use it to like groom themselves too, but like, we're not really sitting around using filiform papillae for anything of that sort. So fungiform papillae. I like to think it's fungi because it's like mushroom shaped. So that's how I kind of kept it straight. So that's why I highlighted mushroom shaped. It's lined by stratified squamous, and this one actually has taste bud, and this is found primarily on the anterior 2/3 surface. So just keep that in mind. Lingual papillae, it's focused on the foliate. Okay, so the foliate, I like to think back to your time when you guys learning about folate, right? Foliate and folate sound very similar. So it's mostly found in children, and it's very rudimentary in adults. It actually kind of semi-degenerates, and it's found in the periphery. So it's like right here of the tongue, okay? Now, your circumvallate is more specifically located posteriorly, right? And it's like dome-shaped. I don't know, like I didn't have a cool way of remembering this other than the fact that there's like this Cirrus von Ebner's gland. That sounds very I don't know, cool, because this has moats. Like look at the moat. Like it sounds like a count living in a castle. So circumvallate papillae sounds like a count living in a fancy castle, and his name is Cirrus von Ebner. He's very serious. He has a moat and everything. So that's how I remembered circumvallate papillae. Parotid glands, this one was surprisingly high yield. This is from your guys' DLA, so just keep this straight. It's classified as pure serous secretions, okay? So if it's serous secretions, it's more liquidy, right, as compared to mucus secretions. So when you guys are going through this, make sure you guys are able to distinguish based off of, one, the buzzwords that are going to be in the question stem, and two, any sort of histological kind of imaging that's given. Obviously, they contain a whole bunch. This is the primary thing that's secreted when you're salivating, right? That it has like a lot of enzymatic components that break down food and other things. So peroxidases, lysozymes, and cystatins, which are often just used to break down food and stabilize your mouth pH in order to deal with incoming food. Okay. Buzzword, intercalated and striated ducts is often seen. Your parotid glands, here it is. It's the longest. If you see the buzzword Stenson's duct, it's the longest excretory duct found in the mouth oral canal. So it opens directly into the oral vestibule and at the parotid papillae. So second molar, it's like located right there, and it just shoots out. Fun fact, if you can actually push up your tongue, and you can actually eject out the parotid secretions. It's a weird, quirky effect and a weird, quirky mechanism. Submandibular glands, important to kind of know because this one has, remember, mucousy components that are brought in. So it's classified as a mixed compound. So what do you guys see? You guys see how it's kind of washed out, right? Because any sort of mucousy products have some amount of glucosaminoglycans, proteins, and sugars. So they get washed out when you're doing PAS staining. So you might you'd have to counterstain with PAS in order for it to show up properly. So excretory mandibular ducts open at either sides of the base of the lingual frenulum. That's a buzzword right there. Lingual frenulum, submandibular glands. That's what I use to get the answer on our DM exam. Now, sublingual, okay, this is a mixed acinite gland. It's pure mucus secretions. So you guys see how a submandibular is a mixed, right? You guys see how it has both mucousy components, which is all washed out, and then you have like the serous component, which are all still like purely stained with H&E. Sublingual, purely mucus, and it will purely wash out with any sort of alcohol staining. So you have to counterstain it with PAS to even fill in these stuff. Most of the times they won't fill it in. So it's a good way to remember that sublingual, everything gets washed out, okay? Here's the distinguishing factors. Just basically run through all this. I'm pretty sure you guys had small group and you had to present these slides. Do these slides. I swear to you, they did show up like 1 or 2 times in each of the blocks, and it helped me a lot that I had went over them. Okay, let's talk about the esophagus. Okay, very simple. Stratified squamous, non-keratinized. It goes through and through, except for when you have, guess what? You have any sort of dysplasia that's changed due to like Barrett's esophagus, any sort of acid reflux. It changes that stratified squamous, non-keratinized, into columnar, which is more glandular in structure, and it helps protect against any sort of like recurrent acid reflux. So important. What you need to remember also is that it produces neutralizing mucus as well from the esophageal cardiac glands. These are not cardiac glands like your heart, okay? Don't get it twisted. Although they might be found more so in the cardiac region of your esophagus, or like more close to the stomach, because that's where you have a lot of your acids kind of springing up. So keep that in mind. Here it is. That's what I was talking about. So remember, it's a metaplasia, dysplasia. It can be reverted back depending on how severe the damage is. What you need to know is that there are cardiac glands that are trying to protect it, but if things go bad enough, it can turn into a cancer. So adenocarcinoma of the esophagus. For Priya, who's in the chat, who's probably getting some real major neoplasia vibes right now. Barrett's esophagus, gastroesophageal reflux. Okay, this is the thing that always shows up with heartburns, right? What happens is stratified squamous gets turned into columnar. I think for you guys, just keep it as metaplasia. Don't add dysplasia to it. You guys will learn later on about dysplasia and proper properties with that in T3 and T4. So just for now, just remember it's metaplasia changes. Okay. 3 types of glands you need to know: cardiac glands, funded gastric glands, and then pyloric glands. Obviously, you guys know your pyloric glands very well. You know your funded glands very well. The cardiac glands are more neutralizing in nature because you don't want acid to basically come up and mess up your esophagus, right? So that's not good. As always, they also like to throw this image in. Do not put intestines. I swear to God, if you guys put intestines, I'm gonna be so sad, because it is not the intestines. These are rugae, are basically the components that essentially allows your stomach to expand as food is brought in, okay? Awesome. Cardiac region, here it is. Pyloric region, you guys know all these things. Short pitted, short cardiac glands, right? Mucus secretion, enteroendocrine function, whereas your pyloric region has long pyloric glands, right? Because remember, long pits, P for P, go together. And they're mostly involved with maintaining your gastric secretions, right? All right, fundic region, you guys know this very well. Isthmus of the glands. The key thing you need to know is that the surface level, right, is basically trying to help maintain and not burn up your stomach lining. Whereas your neck area is working mostly with your parietal cells. And then your fundic, or like the bottommost part of the pit, is where you have your chief cells. Okay, if you know that, you're essentially good for stomach histology. Okay, because they literally will just, let me show you. They zoom in like this and they're like, tell me what these egg-shaped looking cells are. And you have to be able to correctly identify them as parietal cells that are secreting what? It's going to be secreting specifically hydrochloric acid. And those hydrochloric acid are going to take all the things that your chief cells are making and then spit it out into the luminal side of the stomach. Okay, so essentially HCl, parietal cells. Also, you need to know that intrinsic factors. I swear to God, guys, if you guys forget your intrinsic factors, I'm going to be so sad, because it's a very quick way to get a couple of points. Because remember, intrinsic factor allows for your B12 absorption in the distal ileum. Okay, this is why they like to ask anatomy questions compounded with physiology questions, because this is a quick way to test your knowledge of both. What does B12 deficiency present with? And you guys learned this even in T1, macrocytic anemia, right? B12, macrocytic anemia. B12, 12 is a big number, macrocytic anemia. Whereas B6 deficiency, guess what? Smaller number, microcytic anemia. Wow, look at all those wonderful brain cells working to make little random connections that I swear I should have made even when I was going through DM. Okay, mucous cells, important to know, their essential function at the surface level is to produce bicarb and potassium to counteract any of the acid that's there at the surface level, right? Fun fact, H. pylori actually messes around with the mucous cells so that you could potentially get adenocarcinoma of the stomach, something we learned in T4 and we continue to learn till this day. Chief cells, right? They're basically basophilic in presentation. So what you need to remember, right? What are they secreting? Zymogens, right? Zymogens are essentially proteins that haven't been activated. They're basically, they need to be cleaved by a specific enzyme factor. What you need to know, pepsinogen, right? It gets converted into pepsin, one of the important cleaving / destructing factors in our stomach that allows for any sort of breakdown of products being brought in. It's very toxic. It's very powerful. It's one of the things implicated in any sort of peptic ulcers as well. Okay, your parietal cells, you guys already know this, hydrochloric and intrinsic factor, B12 deficiency. If your parietal cells are damaged, right? These are your egg-shaped. I swear to you guys, egg-shaped is a buzzword, or not really a buzzword, but it's a characteristic that can help you identify at the neck level, or like the gastric pit neck level, what is happening. It's also stimulated by gastrin, right? Parietal cells, right? And the receptors for it is H2. Guess what? H2 is a GS coupled system. So something we also continue to learn even in T4. So all this is important knowledge that is compounding. Okay, enteroendocrine stuff is not too important for you guys. All you need to know is that different secretory properties at the lowermost region is basically at the gastric pits are working to put receptors and hormones into the pits to then do the chief cells to make specific things, right? Such as pepsinogen and other things that are going to eventually be pushed into the luminal side. Gastroduodenal junction, very simple. All you need to know, right, with this was they asked us a question about pyloric stenosis. That's essentially as much as it gets. Mucosa, submucosa, and muscularis externa. Run through these histology and do these slides during small group. Peptic ulcers, very simple. If you see this picture, this was literally given to us and they asked us what are some factors that messes around with peptic ulcers. H. pylori, don't smoke, don't take NSAIDs on an empty stomach because they inhibit prostaglandins, and we know all about prostaglandins now, and Zollinger-Ellison syndrome that can lead to gastrinoma, right? Which is an adenoma, adenocarcinoma of the stomach, okay? Small intestines, okay. This is just general properties, like it has like plica circularis, villi, and microvilli, because remember, the end goal of the small intestines is just to absorb a whole bunch of stuff that's coming in, right? So you want to increase the surface area to the max, right? So there's three regions, say it with me, duodenum, jejunum, and ileum. And if you have it straight, you should be good. Small intestines, right? GALT, okay? That's a buzzword. GALT, you need to know your villi as well as your crypts of Lieberkühnen, because these are involved a lot with sort of cancer-like presentations in the small intestines, and you're going to learn more about it. And that's why they spend so much time that you guys learn the fundamentals here, okay? Buzzword, plica circularis, guess what? Small intestines, and you guys should be good. Here are the tissue-specific buzzwords, right? Plica circularis, it's telling you jejunum, okay? If you are seeing any sort of like, you know, central lacteals as well as villi, that's once again, you're still at the small intestinal level. Microvilli, right, are other properties that you need to know and identify, and that's the brush border. Guess what? Same brush borders that are implicated with your lactose and maltose and all sorts of other sugar cleaving components, okay? Intestinal glands, you guys know this, crypts of Lieberkühnen, and their primary function is just to, you guys are going to cover this more in GI physiology, so I'm not going to spend time going too much in depth with it because that's all Dr. Bandelow's lecture. So primary absorptive components, right? They have enterocytes that help bring in stuff. They have goblet cells, right? They basically start replenishing these constantly. They have mucinogens and other things that help maintain a lot of your different gut flora. They have Paneth cells. Paneth cells are a buzzword. You need to know lysozymes, right? Which are antibacterial enzymes. They help maintain your gut flora. And then you need to know your alpha defenses. So Paneth cells, buzzword for small intestines, lysozymes, and alpha defenses. Know those very well, and you'll be good. Okay, duodenum. If you see Brunner's glands, okay, please put duodenum, okay? You're gonna make me very sad if you guys miss this question, because a lot of people miss this, and they unfortunately, they could have gotten a couple of easy points. So jejunum, okay, plica circularis, long prominent villi, they love that. And then the ileum, okay, Peyer's patches. Whenever you see Peyer's patches, ileum, and they literally gave us this picture, that's all you need to know, okay? You're in the ileal region. Celiac disease. So what really happens with cilia is it's the blunting of the villi, because what happens is, this one was a tough one because they never pinpointed this too much, but this is very buzzword heavy. IgA antibodies, right? And it's dealing with gliadin peptides, okay? Celiac disease, you guys got to know this because it can lead to a lot of different presentations that you will see with, guess what? Not being able to process B12. Guess what it's going to lead to? Macrocytic anemia. I mean, obviously you're going to have other issues, but like they tie in a lot of components together with the histology and the question stems. Crohn's disease, okay? This is a more ulcerative form of things. And the buzzword for this is cobblestones. If you see cobblestones, Crohn's disease, C and C go together. You guys should be good with that one. You don't need to know too much about these granuloma formations. That's a very T3, T4 heavy thing, so don't even trip. And it can lead to obstructions. Large intestines, guys. Okay, if they ask you a question on how do you identify the large intestines, Taenia coli, okay? Or you can do a Haustra coli. Most often it's going to be Taenia coli, and then it's going to help you tell. It's going to help you if you follow the Taenia coli all the way down on the right-hand side. You're going to find the appendix. It's another kind of landmark feature. Mucosa, okay, numerous intestinal glands, crypts of Lieberkühnen. Once again, it's important for water and electrolyte absorption that you're going to see with Dr. Bandelow's lecture. There are no Paneth cells, okay, in the epithelia. Keep that straight, because a lot of people put Paneth cells and it's often missed. Okay, myenteric versus mesenteric plexuses. Those are critical. Make sure you guys review those and how and where each of them are located, because it tells you a lot about the GI physiology lectures coming up. Appendix, just memorize this picture and you will be solid. Obviously, they'll give you more than just this picture. So know the positive psoas sign, know any sort of visceral versus somatic pain, and you should be solid, right? You're going to see a lot of like nodules, numerous nodules, and extending into the submucosa. So it's a vestigial organ, so it's going to look very similar to the large intestines, and the Taenia coli is used to identify it. Okay. Anal canal, it's very simple. There's 3 zones you need to keep straight, and the first 2, right? Actually, the first one, colorectal, is above the pectinate line. So obviously, what type of since it's above the pectinate line, what epithelia are you going to see? Simple columnar. They like those type of questions. It is often tested. What are you going to find in the transitional zone? Guess what, guys? Simple columnar to stratified squamous. Very simple. Simple columnar, stratified squamous. It's a transitional zone. And then obviously you're going to get to the squamous zone, and the name says it all. It's squamous. So everything's broken down into 1/3. And you guys already covered a lot of the musculature, because remember, I'm telling you guys, internal anal sphincter, what is it ruled by? Sympathetic, parasympathetic system. They literally threw a picture on there and asked us what type of innervation was running through this. It was a great way to test your ER and your DM knowledge. Guess what? Congenital megacolon comes back even in histology. So which portion is messed up? Is it the dilated portion, or is it the squished, smaller portion? Smaller, okay? I'm going to emphasize that. People keep missing that point, but make sure you guys know that it's the smaller component that's messed up. What are the people basically missing? It's the myenteric plexus in the distal alimentary canal, right? Myenteric plexus is the independent plexus that allows for a lot of the peristaltic movements to happen based off of mechanical changes to the tissue, right? Like basically, any sort of fecal material kind of floats through the system, and it basically takes a smooth muscle and pushes it apart, right? And that process the smooth muscles release certain calcium, calmodulin, and all those things combine together and then allow for the peristaltic movements to happen, whether it's segmentation or propulsion and all sorts of other things. Okay. All right, last thing, guys, hang tight. We're almost done. It's the GI physiology introduction. So obviously, sympathetic versus parasympathetic is what you need to know. That is the extrinsic influence. That's coming from the central nervous system, right? From the brain, from the spinal cord, and eventually going to the peripheral tissues, which in this case isn't really peripheral. It's more so in the digestive system. What you need to know is that it has both sympathetic and parasympathetic. It makes sense, right? When you're running from a bear, you want to make sure you inhibit peristaltic movements so that food and anything that's floating through your system stays there. I would highly recommend you guys review this because a lot of people slept on these lectures because it was so easy, but a lot of our questions came from this, okay? Now let's talk about the intrinsic systems, right? These are independent, somewhat independent from the ANS, and they can function based off of local reflex. What I mean is mechanical changes based off of fecal matter coming in or food coming in or anything. It can cause changes at the myenteric plexus, right? It controls the enteric motility as well as the submucosal secretion, right? Food expands, right? Food expands your GIT and your smooth muscles and your epithelia will basically respond to this automatically and say, hey, maybe I should start secreting out, certain sodium and other products to help break down this food so that, I can eventually absorb it. So there's a lot of different components with this. So myenteric plexus, we're talking about also Orbach plexus, submucosal plexus, right? Mesoners, okay? Keep those straight. Don't twist them up because they often are asked together in question stems. Guess what, guys? Toxic megacolon. Everything's missing. You have no ganglia of the large bowels, absent submucosa, as well as the myenteric plexus, as well as massive distension of the proximal colon, right? That's because everything's kind of blocked up. There's no peristaltic movements here, right? So that tends to be the problem, right? Failure of neurocrest cells, as always. Okay, hormones. This is going to be important. Neurocrine, endocrine, and paracrine are all critical because they all work at the local level, and you got to know all of these hormones straight because, and they have locations for these. I put in a picture and a nice table that outlines all of it. Okay, gastrin is very simple. Guess what? It's gastric acid secretion elevated to the next level. Keep in mind, right? More hydrogen secretion, more acidic properties of the stomach being elevated, whereas somatostatin is the opposite, right? Statins, remember somatostatin? We kind of covered it in ER when you guys came for that review. There's another form that's released from the D cells, from the pancreatic islets and the GI mucosa. So they have questions on the location. So I would suggest you guys make Anki cards and test yourself on all of this, okay? Not so much of the super crazy clinical correlates because some of this is a little too high level for you guys, but make sure you guys go through these. Cholecystokinin, eye cells, right? It's going to lead to, importantly, regulation and breakdown of fatty acids and amino acids so that your body can absorb it in the small intestines. Secretin. This is dealing with bicarb and neutralizing a lot of the acidic components that's coming from the stomach into the duodenum. Remember, multiple things can neutralize acid pH that's being brought into past the pyloric region into the duodenal first and second half, right? This is glucose-dependent insulin-tropin peptide. This isn't covered too in depth, but what you need to know is that it's from the K cells. Motilin is for small intestine. So guess what? When you guys when you haven't eaten in the morning, right? Or like when you guys wake up and you haven't eaten and your stomach is just like still like churning and making sound, that's your MMC producing motilin, right? That little grumbling noise that you hear, it's your body still doing the peristaltic movements, even though there's nothing there. And remember, if you throw a rock in an empty kind of cave, it's going to make sounds. So like, that's what grumbling, or like not grumbling, but like your stomach growling is essentially. Vasoactive intestinal polypeptide, this is dealing with intestinal water and electrolyte secretion so that you can basically coat the lumen with water and other components. But the problem is if you overdo this, it can lead to a lot of watery diarrhea, hypokalemia, and the loss of a lot of your electrolytes, such as sodium and other things. Nitric oxide, obviously you guys have learned nitric oxide since like, I don't know, T1. It's going to lead with smooth muscle relaxation, and it is also important for esophageal sphincter. So whenever you're thinking about esophageal sphincter, achalasia, or any sort of things, nitric oxide is something that's implicated in there. Implicated, and see, there it is. I'm like talking from the top of my head, and it's written right here, right here. Yeah. So NO is implicated in achalasia as well. Ghrelin, guess what? Your stomach growling is it's increased appetite. So that's just a buzzword. It's not actually your stomach growling. So it's just talking about you increasing appetite at the ANS level, because ghrelin actually goes and affects the, when you guys get to NB, they cover the exact location where it messes with. Okay, guys, intrinsic, I swear, please learn it. B12, okay? Macrocytic anemia, it's important at the distal ileum, okay? Very, very buzzword heavy. Gastric acid, you guys know this. Pepsin, right? It's primarily made from pepsinogen, and it's basically, it's a pepsinogen, so it's an O-gin, so it's an inactive form to an active form. And finally, it's going to lead to your pepsin, which helps break down a lot of your products that are coming into your stomach. Bicarb is basically helps to neutralize, and a lot of different hormones are going to be messing with it. Okay, guys, we really thought this wasn't a big kind of component, but we were asked a lot of questions on this. Okay, so you need to cover that table I just covered with you guys, as well as the location of a lot of these cells. Okay, because it's important. Form in our body always often equals function. Okay, so imagine, right, food is coming in, it's hitting the I cells, right? And secretin, and it's hitting a couple of your K cells, right? All of these are going to help deal with making sure your bile comes in, your pancreatic amylases and lipases are brought in. So you want to make sure you cover them based off of function, not just pure memorization, okay? It's going to be hard without context, because I can sit there and tell you guys, okay, know your I cells, S cells, K cells, know your histamine and your chief cells and all those things, but it's going to be more difficult if you guys don't have context for this. Okay, hormonal, regulatory, gastrin, okay? Increases, like it sounds like gastric level, increases gastric parietal cell H plus secretions, okay? Cholecystokinin, CCK, important, because remember, it relaxes the sphincter of Oddi. This is a very big buzzword heavy thing. Gallbladder contractions and sphincter of Oddi. Relaxation, critical, because it allows for bile to then pass from the common bile duct into the second part of the duodenum, and eventually helps, one, neutralize your acidic kind of secretions that are coming from your stomach, as well as deal with pancreatic kind of bicarb secretions. So CCK implicated in all of this. I would know CCK very well. We were asked a whole bunch of questions on it. Secretin, guys, you know the secretin because you guys have covered it. It's an analog to glucagon, so it reduces acidity in the small intestines, stimulates pancreatic bicarb. A lot of things mess around with pancreatic bicarb. It's important, okay? A lot of GI physiology questions that feel easy, but can be challenging if you don't kind of run through this process. As always, they're stimulated by the release of S cells, because remember, S for secretin, S and S kind of go together. CCK is a little bit more challenging because remember, it's a CCK and it's I cell. So you're kind of like, where does the kind of the components kind of come together? Okay, gastric inhibitory peptide, it sounds like exactly what it does. And then glucagon like peptide inhibitory kind of components, they're all going to be functioning at the same level. So what is it doing? Decreases acidic secretions by the parietal cells so that you can limit the acidification of your stomach. And this is happening at a different level, right? Remember, as things are passing into your small intestines, you want to neutralize your acid levels of your stomach so you don't, give yourself an ulcer. So important that you guys know that, and that the factors in the duodenum are critical for maintaining your stomach capabilities, right? The stomach acidic capabilities. Paracrine components is important. Okay, somatostatin, need to know very well. It inhibits gastric hydrochloric secretions, as well as histamine. Okay, histamine is very, okay. So if you guys are stressed out, right now your blood pressure is high, you have a whole bunch of histamine floating around, and this is actually one of the ways, the mechanisms at the local tissue level, it can lead to peptic ulcers. Okay, that's going to essentially result in a lot of problems with, that's why a lot of people get ulcers. Okay. So don't try to de-stress once in a while so you don't build those ulcers and mess yourself up. Neurocrine regulatory, okay, VIP. It's not like the VIP that you would think of. It's vasoactive intestinal peptide, and it's basically helps from the neuronal level, right, from your ANS. It limits secretion, right? It limits the acidification via secretion, right? Similar to a lot of different things that you see in the local systems. Gastric releasing peptide, GRP, also known as bomzizin. I have no idea how to say that, but it's from the G cells, okay? G for gastrin releasing peptide, makes sense. Enkephalins, you guys actually know your enkephalins from, I believe, like pain lectures that you guys might have covered before. So just keep that in mind. Gastric parietal cells. What did I tell you guys about H2? It's your GS, okay? Guess what? You actually have to learn simantadine again. Actually, Priya and I, we just finished covering it for one of our exams. So learn these one more time. And I'm telling you guys, these are important. You can also use a couple of things to help maintain your gut lumen via somatostatins and your prostaglandins, because they have a GI property, not GI as in like your gastrointestinal. It's like GI, inhibitory cyclic AMP, and then that's going to decrease your amount of hydrochloric acid that's released. Okay. Now, they can ask you questions here on any level. This was actually very high yield diagram that I myself thought, there's no way they're going to ask this much detail, but they did. And what you need to know are the drugs, omeprazole. You need to know your receptors, right? And you need to know each of the components here. You don't need to spend too much time on the drugs because they will be covered again. But just good to know your atropine and good to know your simantadine, and it's good to know your omeprazole, and you should be good. Central appetite regulator, guys, you know your leptin. It's there. Your leptin is just your satisfactory thing that's released by the fat cells. So if you have more fat cells, your satisfactory hormone kind of like component is going to be more so regulated. Ghrelin is the hungry one, and that's from your gastric cells. So it opposites like leptin. So you'll see it in the ANS lectures as well. So don't trip. I'm putting this again because I want to emphasize that you guys should try to memorize this, but also work through the functions of it. Okay. As always, I included some external resources. I wanted to make sure you guys looked at some of these, because if you guys have some difficulty with the more finer details, they're here, right? Especially for biochemistry, I really like Dirty Med because it gives you a nice overview and gives you those details that you need. Guess what? If you just click on the link that's in your PDF right now, it would take you to those specific websites. And I've already curated all those playlists for you guys to go over. And I was kind enough to just link you guys out directly to your McDaddy 700 question series, and you should be good. Let me stop recording. Thank you guys for coming. I'll say a couple of minutes after and answer any questions that you guys have.
Thank you.